Good morning, this is RV Falkor on the ROV dive 398. This is a dive at in front of Noddy Reef on Cape York Peninsula, and this is our sixth dive for this expedition called the Northern Depths of the Great Barrier Reef. We are on the eastern side of Cape York Peninsula, and Cape York Peninsula is the focus of this expedition. My name is Dr. Robin Beeman. I'm from James Cook University, and we're joined on board by a science team of Marty McNeil from Queensland University Technology, Vicky Lowe from University of Queensland, Valerie and Joan as postgraduate students from James Cook University. Assisting here with the ROV pilots, we presently have Cody and J-Rod, including James on the wireless, on the Wirecast, to help us go live here. So we welcome you all aboard for this this ROV dive 398, and it'll be we hope it'll be a really interesting dive that we've got planned for you today. Our main interest here is we're at the very head of a canyon that is directly east of a reef. This is Noddy Reef. It's very close to us. It's only a few, 1 or 2 kilometres just west of where the ship is presently. And the idea is that we will run the SuBastian ROV up the steep head of the canyon from around about this 820 metre mark up to the base of a steep cliff. It's around 720 metres depth. We'll go up that steep cliff to a ledge which is around about 500 to 450 metres at its easternmost point, and then we'll run across this ledge towards the base of the Great Barrier Reef. And it's quite clear that we're looking at the Great Barrier Reef as we understand it. This layers of limestone strata starts around 200 metres depth and then goes all the way up to sea surface, and we can see that quite clearly from we can see the reef quite clearly from the back deck of the Falkor. So we're going to pass through this cold water coral zone. We will be doing sampling throughout this particular transect, and we've just collected two push cores. These are sediment cores. We do it every ROV site if we can, and it's the idea. The idea is to get ourselves a good sample of the sediment at each site, so we can characterise the seafloor across all of the different places the SuBastian's visiting on the Great Barrier Reef margin. And I have Marty McNeil sitting next to me. Morning, Marty.
Morning, Rob. Good morning, everybody at home, on shore, and wherever you're joining us from around the world today. I'd just like to say good morning to the ERB 102 Evolving Earth class at QUT. Good morning to all of you. Good morning, Luke. Luke was on leg 1 of this voyage and will be joining us again for leg 3. So stay tuned. It should be an interesting dive today.
Yeah, that's right, Marty. A lot of diversity here. So, we originally planned this dive so that it was mainly mesophotic. Mesophotic being anywhere probably 400 metres and shallower, certainly within that photic zone, twilight becoming photic zone. But when we actually looked at the depths, it's such a dramatic, steep drop-off here. It's like a huge bowl. That it was quite easy to get down to 800 metres at the bottom of our target depth, but because we have these very, very steep rises, like almost cliff-like faces, we're going to cover a lot of vertical ground today. So already we're seeing on the seafloor rubble. And because the coral reef is so close to us, it's very likely this is shallow coral that's rolled down the steep slope and is lying at these lower slope depths. Ooh, what have we got here, Marty? Is this a polychaete or a holothurian? Hatchet fish.
Holosurian.
Just a hatchet fish has just
Stealing
jumped
the
into view.
show.
I was looking down on the seafloor, something
Yeah.
wriggling around here.
At first glance,
And-
I thought Holosurian.
And over on the
There's
right
another
too,
one over on the right.
small pink. We'd have to
Yeah,
get a little
another
bit
one.
closer.
Yeah, there's a few.
Yeah, so we're thinking perhaps sea cucumber or even polychaetes. You know, there's certainly the seafloor is made well use of polychaetes. Most of them are infaunal though, but you can get flatworms and the like that can wriggle around on the surface. So we'll get a close look. We've only just got on the seafloor about 5-10 minutes ago. We've taken our first push cores, so that done, we can start having a bit of a wander around.
Good morning on the YouTube chat to Dougal Lindsay and Will White. Nice to see that you're out there joining us today.
Yeah,
A couple of our collaborators that are onshore at their respective universities. Well, Will's at CSIRO in Hobart and Dougal is at Tech in Japan. Both principal investigators are involved in this voyage, but remotely. They weren't able to come and join us on the ship.
that's right. It was quite challenging to get people on board. So we've only got a small science team of 5 at the moment. Ably assisted by a professional crew of the Falkor, of course. But we have a very large number of principal investigators watching from all around the world, and I think we're over 15 now. People who are really directly involved with decision making, the working up the samples that we're collecting, and also the vision that we're collecting. So, very large team ashore, which of course is quite typical of any modern research expedition. The aim of the game is collaboration, and so the more people you can bring to help work up the data and imagery, the better. And this is how science advances, by bringing as many people, as many eyes
That's right.
as possible.
That's what we call multidisciplinary research, or a multidisciplinary approach. It's such a rare opportunity to have ship time that we want to maximise our data collection and sampling, not just for ourselves and our immediate research groups, but there are also many collaborators that we work with all around the world, and we can use this ship time to collect samples and data for other scientists and other research groups, and that way we're really getting the most out of the time that we have aboard RV Falkor.
This looks like a black coral to me. Has it got these twin paired sets of polyps?
You can hear good meeting going on behind us here.
That's right, a bit of a collaboration.
Discussion.
So we also have, we're also reaching out to our colleagues through Slack. We've got a Slack workspace, and we can directly communicate with each other.
Sitting on something hard.
Yeah, so you talk about multidisciplinary, Marty. I'm just going through a mental list of all the things, all the different disciplines. So straight up we have marine geology,
Yep.
and that stretches out into all sorts of things from deep water carbonate geology through to shallow coral reef type environments, and including Yusuke Yokoyama, who's really interested in age dating. So he has a real speciality in geochem type work.
Geochemistry, isotopes, trace elements, and then geophysics is a different discipline in itself, but you could almost bucket it with the geology.
Yeah, I tend to be more the geophysics, so I'm more into the sonar and the mapping. So certainly all of the mapping data that we're collecting is of incredible use. It's the fundamental data set from which everything is kind of compared against. So that's more my field. I guess an acoustician, perhaps someone might call me. But when we think of like oceanographers, people are interested in the temperature, salinity, density of the water column as we're travelling through these waters. So the oceanographers, there's that discipline. The plankton specialists like Dougal are involved. So mid-water, it's not an area that I really had spent a lot of time in. We tend to be pushing straight through to the seafloor.
For me, it's been a real highlight to see that aspect of the research. And then, of course, there's Benthic ecologists and biologists.
Yeah, the fish has been really fascinating. We don't have any fish biologists on board, but we certainly are being really well helped by everybody ashore, experts and non-expert alike. We really appreciate that.
Yeah.
I call them the fish army, you know. But, you know, just the mid-water, there's mid-water fish.
Yeah.
And of course all the demersal or benthic fish as well. It's just a huge range of, huge diversity of fish. That's been fascinating. Who else are we, what other disciplines are we dealing with here?
Well, even within oceanography, there's the physical oceanography and chemical oceanography, which we've kind of touched on. Taxonomy, so
yeah, a lot of that.
What we're collecting for right now.
That's right. That's a big part of this. So it's not just about mapping the seafloor and getting good imagery. We actually do have to bring samples back, and this is quite important for people that work in museums, where you get type specimens. You can look at samples under a microscope and really classify them into their proper biological tree, and that way you can tell exactly what species you're dealing with.
That's right. So this, I guess, I'm assuming it's a black coral, is that the word? Yep. Yeah. This black coral that we've just collected is for a genome project that our colleague and collaborator Jeremy Horowitz at James Cook University in Townsville is involved in. So this sample will get tucked away in its own little pocket on the ROV, and
this is a genome project one?
Yeah, this one's going to go into the separate coral quiver.
Yeah, now this is with a project with Jeremy Horowitz. He's and Tom Bridge working with the Sanger Institute, I believe, and also the Smithsonian Institute. So these are full genome DNA sequencing of deep water corals and their symbionts.
Yeah, they're associates.
They're associates, be they, you know, algae or bacteria or maybe even small crustaceans, to try and understand how perhaps they've co-evolved. Yeah, it's quite interesting. Jeremy obviously knows more about it than we do, but what we have to do on board here is we have to preserve these samples as best as possible.
And as quickly as possible.
Yeah, and we're doing this through flash freezing with liquid nitrogen. So we have a big bottle of liquid nitrogen on board, and Valerie and Joan had their first go at it the other day, and it all went quite successfully.
Very smoothly, yep.
Yeah. But we are talking seriously cold temperatures here. Yeah. Sub minus 100, right?
What is the temperature of liquid nitrogen? Someone will be able to tell us.
Do you know how cold that stuff is?
The liquid nitrogen?
Yeah.
Colder than negative 80 degrees. Minus 179.
Wow.
So well sub minus 100 degrees C. It has to be flash frozen, and then of course we have to get it ashore. There's quite a lot of logistics involved around this.
Yeah.
All right, we might we'll head off pretty shortly. So we've just been sitting here for about 15 minutes, just getting our first push course samples and already our first black coral sample. What's underneath here, Marty?
A polychaete sitting underneath.
Yeah.
There, looks like a polychaete.
Yeah. There's that kind of segmented look about it. The seafloor strikes me as fairly rubbly here. So I'd say we're going to see a lot of this coral rubble, and it'll only increase as we get closer and closer to the reef through the dive. What have we got here? Is that a little sea cucumber? Oh, here we
Here
go.
we go.
What's this? This is a sea
Swimming
cucumber.
cucumber.
Yeah.
They're bizarre, aren't they?
Yeah, yeah. Now we do see them swimming, of course, but this is a little bit different. These small pink ones. We've often seen these large purple Benthic, I think, is the genus.
Yeah, they're quite big, aren't they?
Yeah, I'm not sure what this one is. We did reach out to Melanie Mackenzie at Museum of Victoria, who's a sea cucumber specialist, to help us ID these animals. We aren't sampling them, so we're just going to have to just use whatever vision we can to reach out to other experts. So I guess this is the beauty of YouTube and the like, that we can actually reach out to all these other experts, and we have this beautiful vision. It's been collected in 4K on board, and we can get still image shots of them. So really, really nice, sharp images. Okay, so small pink type sea cucumber.
Yep. Start moving.
All right, so we're now going to track. Oh, is a jelly just drifting into view?
Top left.
Yep. Dougal, hope you're paying attention in Japan. That's very pretty.
Let's just give
Just drifting past.
I
Not
bet-
too much swimming going on here.
that is tiny. Just waiting for the delay to catch up on the YouTube so that Dougal Lindsay says, "Wow." I could hear your voice in my head, Dougal. Oh, what's that?
Can you read that out?
Benthic hyalinus, question mark.
Trick is to say.
First record in the southern hemisphere outside of Antarctica, possibly.
Wow. Okay. Definitely got a wow from that.
There you go.
So the
Another Falkor
trick is
first.
to say it fast, Marty. That way no one knows you can't mispronounce it.
I don't think I'm fooling anyone.
Oh, what do we got here? Is this an echiuran worm? Something long worm-like on the seafloor here. And we've got some fish coming in. We saw some hatchet fish on the way down, around about 600 meters depth. So they're, really, really prominent shape. Now this is an echiuran worm. Okay, would you mind just touching this? Thanks, J-Rod. Yeah, just to show people, the actual animal is on the center. The proboscis is sort of split in this Y shape. If you just give it a boop, you'll see it disappear into that hole. There we go. There we go. Starting to retract now.
It's pretty slow this morning.
Yeah.
It's not even Monday.
Yeah, it's a good size though. I mean, that's 10 centimeters across the lasers. There we
There
go.
it goes.
And there's a couple of sea cucumbers off and running too. So these echiuran worms lay out this long proboscis, and we've seen them meters long in places, and they're collecting food. I don't know if the food is actually passed down the proboscis while it's stretched out, or they just wait until food sticks to it and then they withdraw it into their mouth. Maybe someone ashore can have a look and see how what their mode of feeding is. Getting a lot of sea
Lots
cucumbers
of those
here.
little small pink ones.
Yeah. Yeah. Lots of coarse rubble here, Marty. I mean, we know it is quite muddy. We've taken our push core, and there's a lot of mud there clearly, but there's a lot of rubble here as well. Some bigger blocks. All right, so we're going to just slowly make our way up slope. We're actually in the head of the canyon.
Check that out.
We're currently at 811 meters.
Zoom in on that.
Is that a little shrimp? Oh, and there's a flounder. There's a little flatfish just to the left of it. See down.
Yep, the lasers are on it right
Yeah,
now.
right over the
10
lasers.
centimetres.
You can just see his eyes. Yeah,
Well hidden.
yeah. Very well camouflaged. On our last dive at Tidman Knoll, we did see a lot of flounders, flatfish. They all had that really distinct colouring though.
Yeah, that's right.
This one's nearly completely masked by the mud over the top. So I don't know the difference between a right eye flounder or a left eye flounder. Is it one is they're looking in that direction, or is the eyes over on the other side? I don't know.
I have no idea.
Maybe our fish army people can tell us. I just know them as a flounder.
Yeah.
Yeah. Oh, what have we got here? Something down the bottom, perhaps a little hermit crab.
It's interesting.
No, it's a crab with shells with
It's
and
got
coral on it.
it.
It's yeah, one of these ornamental crabs.
Great crab.
But it's got.
God.
We're
That's
looking at
ambitious,
coral.
isn't it?
We could see the
Good
coral
grief.
polyps. That is quite a load.
Wow.
That is one hell of a headdress, don't
Isn't
you
it?
think?
That is fancy. Man, this guy's
How
struggling.
much weight has that thing got on its head?
Good grief.
That would win the ladies dress up stakes
Wouldn't
at
it?
the
At the
Australian
races,
races.
yeah.
Yeah, you
What
line
do they
them
call
up.
it? Fashion on the field?
Fashion on the field. Yeah, this one would definitely win it in the Fascinator stakes.
That can't be easy to move along like that.
Because these are scleractinian corals. This is calcite, calcium carbonate.
Yeah.
This is not. Yeah.
Wow. He's burning up a lot of energy.
Whoa! There we go. Have a bit of a, and we got a little squat lobster to the right here. Yeah.
So we've had a request from one of our onshore collaborators, Merrick Ekins, who's the sessile invertebrate curator at the Queensland Museum, and he would actually like to collect that. Not for the crab, but for the zoanthid colony that's on it. So, yeah, I think we'd like to collect that, please. If you can get him, he's not too quick for you.
Yeah, he's quite small.
The crab is not sessile, no.
Yeah, it's disappearing quick.
Oh, head him off at the pass. Okay.
Okay. So just on sampling, we are very careful about what we take. We don't want to overload ourselves here on the ship, but we have permits at various zones within the Great Barrier Reef Marine Park. And this is worked out, months before the expedition starts. And we reach out to the various taxonomist biologists who want samples. So we have like a wish list of things that we're after. So we're not collecting anything and everything. We're being very circumspect about what we're collecting and why we're collecting it.
There it is, it's quite small.
Yes, Will. Yeah, see what you're saying about that flatfish. It was quite masked by the sediment, but I'm not sure if you're watching the previous dive, the flatfish there that ran that Tidman Knoll. They all had really distinct markings, really all of them had the same markings, so it probably wouldn't be too difficult to find out what species of fish they are. But be interested to know this right-eyed fish, left-eyed fish thing. How do you determine that? Pretty muddy here, Artie, but still a lot of rubble in amongst it all. Yeah. Yeah, thanks for that, Will. Just reading through your explanation. And I'm not sure if you noticed, there was some hatchet fish around us as well. We saw more of them up in the water column, and they were quite obvious.
Thank you. I could go on A with the sponge. Thank you.
Thanks very much, gents. So part of the whole archiving process is that we take, we've got an event logger here, so we've got Valerie on the event logger behind us, and so as the animal was collected, we take a photo, it's brought up to interview, we take another photo, and then it's photographed into the box. So there's a process that follows the sample all the way through until it's stored in ethanol, and then it's bagged and eventually taken ashore. So it's quite an effort to make sure we don't lose track of what we've collected, where we've collected, and when it was collected. This little brittle star on the right. There's a lot going on in the seafloor here, isn't there, Marty?
There's a lot, yeah. I think we need to go and look at that.
Yeah, that's what you want, right?
I think so, yep. Oh, hang on, maybe not.
It's a fair way off.
Or was it that one? Might have been that.
Yeah, it was just over here on the right. Yeah, good. Oh, something attached to it. Oh, a little
crustacean of some sort.
amphipod, perhaps? Yeah, it looks like a little amphipod. So the isopods, I think, you know them, they're flat, they're the pill bug style, and then the laterally vertically narrow ones are the amphipods.
Okay. This looks interesting.
Yeah, I don't know what that is. I mean, the colony looks a little. It's very very delicate. Yeah, let's just have a spin around to the right here. Something hanging off the edge of it.
Yeah, this one.
Okay, what's that? Is that a
ctenophore?
Oh, it's a Benthic
ctenophore.
ctenophore. Yeah, now these can be very pretty. So Dougal, maybe you could just write a little bit about what a Benthic ctenophore is. So they're a comb jelly, obviously, but these are actually attached to the seafloor. And they have some really pretty colours. can be even quite big too.
Yeah, we saw quite a big one on a previous dive, which I can't even remember now if it was on this trip or the or the last
yeah,
trip.
too many
the last
Falkor
trip.
trips for you,
It was the
obviously.
last trip.
Yeah, no, it was the last
And it
expedition.
was quite big, and I'd never heard of a Benthic ctenophore before, so
Yeah, strange. Like rabbit ears, you know,
Yeah, yeah.
like a big jelly with rabbit ears. Very colourful.
I can't even say that. Laurustis, attached by mouth to its substrate.
Taps, okay.
Zoom in on the internal canals to see the branching structure.
Yeah.
Long piece hanging out of it. See that?
Yeah.
Oh, yeah.
It's got filaments or something hanging out of it?
Yeah, or it's tentacles,
tentacles.
however it's feeding. Are these feeding tentacles, Dougal? You can just see them waving. Yeah, it's very fine. It's like a very fine hair that's hanging off the lower lobe, and the hair, it's the tentacle itself has got smaller tendrils coming off the longer tentacle.
Yeah, Dougal says, yeah, feeding tentacles.
Okay, we're just trying to adjust the lighting here. Yeah, side branches, that's how it looks to us too.
So it sounds like this is a baby from the description on the YouTube chat.
It's got really strange internal organs, hasn't it? These sort of canals, these white, this kind of this loop of white on the outside. Are these the filaments, like the swimming filaments they do?
Oh, the cilia.
The cilia, yeah.
I have no idea. I think we're going to go back and take another look at this. Yeah, at that.
Look closer to this, and we'll go back to that.
Yep.
So we can zoom in.
Sure.
Yeah, we're just going to manoeuvre the SuBastian here.
So Dougal says that those whitish structures were reduced comb rows.
Okay.
Yep.
Okay, so you can kind of make out that it's a ctenophore comb jelly. Yeah, I remember the first time I saw it, I was like, what on earth is this?
Yeah,
Like,
yeah.
so I've seen lots of comb jellies swimming in the ocean, but you know, nothing that's attached to the seafloor.
Yeah.
But very pretty, you know, lots of different colours, and can be quite large. You know.
Similarly with that benthic siphonophore that we saw the other day. Who knew?
Yeah.
I didn't know there was such a thing as a benthic
Yeah.
siphonophore.
Who invents these things?
So
eh?
we saw it because we see them in the water column, the siphonophores.
And then you see one on the attached to the seafloor, and yeah, completely crazy.
So we're just going to get zoomed in on this and take a good close look at it and see what we've got here.
And a shout out to my brother Greg, who's watching from Kia Ora in Sunshine Coast of Queensland. Great to have you joining us there, Greg. So we're in a place called, we're just seaward of Noddy Reef, and Noddy Reef is one of these outer barrier reefs near Princess Charlotte Bay on Cape York Peninsula. So if you're not familiar with Australian geography, if you have a look at Australia and say Google Earth, that big long pointed peninsula on the northeast side of Australia, stretching up towards Papua New Guinea, that is Cape York Peninsula. And Cairns is probably the main town closest to Cape York. This is where I live with my family, and where leg 2 started from with the Falkor.
That's right.
But everything north of Cairns is essentially Cape York Peninsula. It's around about 700 kilometres north to south, so it's quite a big chunk of land. But importantly, it's considered a bit of a frontier area. There aren't a lot of people living. Oh, there's fish. Good size too. So
yeah, we're still we're still debating this potential coral / potential sponge. Maybe I'm just gonna tag Merrick. I hope he's watching.
Yes, if Merrick is watching, he's interested in carnivorous sponges. This is a type of sponge where they have sharp little spines on the outside, and they basically spear. They spear animals and are absorbed into the flesh. So quite strange and fairly delicate looking, like this particular one.
I think we are going to collect it either way, but we're just trying to decide about which project it's going to go to, and which will then dictate where we put it on SuBastian.
Yeah, it'll either be flash frozen with liquid nitrogen, or just put in the bio box, and those ones just get preserved in ethanol.
It was on a minute ago, but it's not on now. So maybe we'll just collect it and put it in a bio box. Oh, is it as close as you can get, Cody?
Well, if I pick it up, I can read right.
Yeah, if he picks it up.
Yeah, let's do that. Let's do that.
Just closer I can get there.
Yeah.
Very translucent. Sitting up. Very spongy. Very spongy.
Yeah.
So we're getting from Noonie, Neoscopolis. It was that large silvery fish that drifted past us. Obviously attracted by the lights of the ROV. So we're thinking this might be a carnivorous sponge. We're going to bag and tag it. But just going back to the geography, so we're on Cape York Peninsula. The area on the land area is actually considered quite a frontier zone. It's not very populated compared to the rest of Australia. There are Aboriginal communities, some mining towns, a few support towns, some cattle stations, and quite large national parks because of how unique it is. In the offshore area, similarly, it's considered a frontier zone. Very few people get up here. Certainly in the offshore deeper waters, there's been no systematic mapping prior to the Falkor arriving, and there's been no deep ROV dives ever in this part of the far northern Great Barrier Reef. So there's a lot of firsts here. We're getting amazing 3D seafloor mapping data, as well as having the opportunity to dive with the SuBastian ROV.
Okay. That's great. Thanks, J-Rod. We'll collect that and put it in box 1A.
1A.
So hang on. Do you want to call it?
Yeah, flash freeze.
Well, Merrick. For Merrick? Is the flash frozen?
Yeah.
No, Noni, we don't have any crustacean experts aboard, nor do we have any identified yet within the principal investigators, the big group that we currently have. But we're certainly open to crustacean experts getting involved with this. We do know people at other various museums around the country, and this is where we have to really reach out and use our network of principal investigators to reach out to these other experts. And that way, we get the most benefit out of all of this imagery and the samples that we're collecting. There's quite a lot hanging off this, isn't there, Marty? There are little amphipods, maybe a few crustacean. Looks like a worm or two. There's quite a bit on here. If it's carnivorous sponge, maybe they're being eaten.
Sorry for the delay here. We're just waiting on a decision about where we're going to put this.
Yeah, it's in the fruit box, and they're doing the same project. America and Germany doing the flash freeze, the same project.
Okay. Right.
Alrighty.
So do you want it to go? So does it go in the same quiver with the other sample or in the other quiver?
Alright, so it's going in
Uh,
in a tube.
two. Yes. Yep. In a quiver.
Okay, this one's going to be flash frozen. So we're going for the full genome sequencing of the animal and its associates. So these tubes here, we're calling them quivers, you know, like an arrow quiver. And what's important about it is we can seal them up quite well. That way the sample doesn't get, what would you call it, just contamination.
Degraded.
Yeah,
the
you-
DNA gets degraded. If
Yeah, we don't want it to be contaminated with any other samples, so it'll go into its own quiver with a seal put on it. And then when the ROV is pulled up at the end of the day, we go straight out to these particular quivers, and they're the ones that are flash frozen. And just like to welcome Julia Summerling, who's joined us on YouTube. Julia, you've probably sailed through these waters a few times. Julia's probably done more diving in the remote parts of the Great Barrier Reef than perhaps anyone on Earth. So Julie, we're just a place called Noddy Reef. In fact, it is an unnamed reef. It's just, we're calling it Noddy Reef because its closest named reef is Noddy Reef. It's north of Prince of Charlotte Bay near Creech Reef. So that's quite a prominent reef. It's sort of just northeast of Princess Charlotte Bay. And Princess Charlotte Bay, if you're looking at a map of Australia and you've got your eyes zoomed in on this Cape York Peninsula, this long triangular pointed peninsula, there's a very, very large prominent bay on the about halfway up it. Now this is Princess Charlotte Bay, and we are just northeast of this bay on the outer edge of the Great Barrier Reef here. So Cairns is approximately 400 kilometres due south of us from where we are now. Oh, here we go. There's an inset. So the grey blue is what we've been mapping over the last week, and you can see how rugged it is. These are these big canyons that drain the edge of the Great Barrier Reef. This is just a 3D depth model of the whole GBR. The red and yellow areas are the shallow coral reef that's been previously mapped by airborne LiDAR conducted by the Australian Hydrographic Office and the Navy, and we've been mapping all of that steep area to seaward of it, the offshore Cape York area. So this has never been done before. It's the first time it's been revealed, and you can just see the ROV transect is right in the middle. We're in this big bowl-shaped canyon. This is the head of a canyon that's draining down into what's called the Queensland Trough. It's about 2 kilometres deep at its deepest point. That's just east of where we are. And we're just at the start of this ROV transect, and we're going to start walking our way up the wall. We're going to go up this very, very steep cliff. It's a good 100-metre high cliff, and then onto a ledge, and finish the dive at the base of the Great Barrier Reef, the base being where the limestone starts, which is around 200 metres depth. So back in geological time, when the Great Barrier Reef first started to lay down all its different limestone layers, about 200 metres depth. And so we're doing this transect up this quite interesting topography today. We're only at the very start of this dive, so we're still at the base of this really steep cliff. And thanks to James. James is on the Wirecast doing all of the live YouTube and the insets. So pretty important team, a lot of concentration required to pull all this together. The ROV team, of course, the bridge staff, they're up there on the bridge. They're about 3 decks above us, making sure the ship is held in position through dynamic positioning, pointing into the wind, looking out for conditions all around us. So it's quite a delicate dance by a big team of people, both the ship and, of course, the ROV on a tether down at this 800 meters depth where we currently are. That's right, we're just going to have a closer look at this benthic ctenophore. So we're going to try and get as shallow as possible. I'm thinking maybe 100, maybe 60 meters depth. A lot depends on the weather conditions. It actually is fairly windy where we are, but the currents are really good. Like we only had like 0.3 of a knot, so the currents are not a problem. It really is down to the wind, which is blowing around about 22 knots at the moment, so it's a bit strong for my liking, but we'll just see how we go through the day, and we'll bring the ROV up as high as we can safely do it.
That we're at max zoom?
Yeah, that was max.
Yep, that was max.
Yeah, max zoom. Here we go. What do we got? Another squat lobster or a hermit crab? Yeah, another hermit crab with the soft coral or coral on its back.
Hobo crab. Okay. Carry on?
Yes,
Yep,
off we
on
go. We
Thank you.
have quite a ways to go yet. We're only really at the start. We haven't moved probably very far at all. But yeah, lots to see, obviously, just in the short distance we've run.
We went a total of 10 meters.
We've been a total of 10 meters, and we've already got quite a good sample collection. But we've got nearly 800 meters vertical height to cover today, and the horizontal distance is around about 1 kilometre. And lots of physiography. So we've got this quite steep wall ahead of us, which starts at around 700 meters and it climbs up to 500. So it's about a 200 meter high cliff, and then we run across this ledge to the base of the Great Barrier Reef, where the limestone strata starts to build up. That's at about 200 meters, and then we'll go up as far as we can safely climb. So lots to see today. We're in the cold water coral zone. It's about 6.1 degrees Celsius. This is a gooseneck barnacle. This is a barnacle here, guys. Can we just have a closer look at this? Oh, I've gone past it. Yeah, it was a gooseneck barnacle. Yeah.
You see that comment on Facebook? That was a Google Maps.
Is that a great purpose?
Yeah, this is gooseneck barnacle here. Just withdrew its feeding, not tentacles. It's feeding appendages, its legs, right? So it doesn't look like much, but it's live. It's a crustacean, right? So this is what it doesn't look like a crustacean, but it really is. Of course, when they're free swimming, when they're larvae, they look just like a small crab or shrimp. But when they settle, they create these kind of leathery tubes, which is the neck part of it, and these hard calcareous plates.
Yeah, that's right.
And there's about 5 or 6 plates, and they join together and they open up, and the feeding legs come out and filter feed. They're filter feeders essentially.
Yep.
Yeah, and when the animal dies, the plates break up, and you're often left with these strange, almost fingernail-like plates left on the seafloor. Thanks, gents. Yeah.
Okay, let's keep going.
Yeah, we'll keep going. Yeah, so people asking where we are. If you look for Noddy Reef, now Noddy Reef is a named reef. That's on the middle shelf of the Cape York Great Barrier Reef. If you go due east of there to the barrier reefs east of Noddy Reef, that is the reef we're on. It's quite prominent. It has this really distinct bowl shape on the seaward side of it, and we're diving in the head of the canyon of that unnamed reef. And here we have another Echiurine worm. Yeah, so shout out to Chris Jones from the Great Barrier Reef Marine Park Authority. Chris has been following our dives and our adventures for a long time now. Oh, we've got a lot happening here. We've got a sea cucumber, big with really long, what would you call those?
What do you call appendages?
The appendages, long appendages. We have a fish, and this is an Echiurine worm with this distinctive Y-shaped proboscis. It's a busy site, isn't it?
Yeah.
We've only been 10 metres, and there's just so much on the seafloor. So one of the other sea cucumber we've seen is this smaller pink sea cucumber that was swimming around before. And this one's obviously quite different.
I think we want to keep progressing forward.
Yeah, we need to
Don't
keep
wanna spend
moving
too much time
upslope.
on these things.
Yeah, we got a fair distance to cover today. There we go. There's the small pink sea cucumbers we just passed over. And there's quite a few of them actually out here. So yeah, like a long Echiurine worm. Seem to be very common in in these deeper waters. We've seen them on rocky environments, fully sandy environments. Oh, look at this! Lots of sea cucumbers. And we're fortunate to have Joan on board, part of our science team, a postgraduate student from James Cook University, and she is our resident sea cucumber expert. Xenophyphores as well. These little golf ball sized, these are called tests, is the technical name, and these are xenophyphores. They're a single celled foraminifera. And if you want to learn about them, on the previous expedition, the Seamounts, Canyons and Reefs of the Coral Sea, Rachel Pryslowski wrote a great little blog article about these xenophyphores. And they're really fascinating. They're found in organic rich environments, so bases of coral reefs where a lot of organic material fall down. And we've seen them very common in the deeper oceans. They're quite delicate. The structure is actually made up of glued mud, isn't it?
Yeah, agglutinated.
Agglutinated mud.
Is the term that they use for it.
Yeah, that's a nice looking shrimp. Look at the antennae stretching back.
They're long, aren't they?
Yeah, it's a good size, good, over 10 centimetres long. Oh, we've seen that. What's that? But the xenophyphore, we actually collected one, and then we scanned it with our cameras and created a 3D model of it, didn't we?
That's right. Yeah, we created a 3D photogrammetry model, and then we brought that into another different piece of software and 3D printed it.
That's right. We have a very big 3D printer on board here. For the ROV team, they're often using the 3D printer to create parts for the SuBastian, so bolts or plates, or they can pretty much make anything here. We've got J-Rod, he's our 3D printing expert, and anyway, we gave him a 3D model of that xenophyphore, and I've got it sitting on my shelf back at James Cook Uni. Fantastic. I can hold a piece of, I can hold a life-size xenophyphore in my hand. Because the actual animal, it's very delicate. It's quite easy to crumble. It's only made of, I guess, mud all glued together. So if you pressed on it, it would just crumble. anyway, we managed to keep it in quite good condition so that we could photogrammetry scan it. So this is becoming a new kind of tool, scientific tool, doing photogrammetry or structure from motion. People are
all
doing it
sorts
with
of
coral
applications.
reef. You know, you can take a piece of coral and scan it and get really great detail, you know, great three-dimensional detail on the polyp structure and how the branches join together. People are starting to use it for scientific purposes like understanding, I guess, habitat, space, rugosity and things like this.
So we'll be doing a little bit of a changeover in the commentary chair shortly. Rob and myself will be stepping out to go and join a ship to shore call with some students, university students from UC Berkeley. So we'll be stepping out shortly to get that set up and underway, and we'll be handing over to the rest of the science team here. I think Valerie and Joan are going to take a turn at chatting with you through the dive. And Joan will be able to tell us all about holothurians. Is there a sponge on that? What have we got on here? Got a sponge on the edge there? I think so.
Christopher, you want to jump in? Sure.
So we're just doing an ROV team handover from Cody to Chris. I've only got an ROV team of 3 on board, so with a long dive today, the guys need to change out frequently because of the concentration required to drive it.
I think, no, carry on.
We're going to head off?
Yep, they're going to head off. We're going to head off.
Yep, so we're handing over to the other science team. Marty and I are going to do a ship to shore with Berkeley University for about an hour, and we'll join you at the end of that.
So thanks, Rob and Marty. They are going to do the ship to shore lecture with UC Berkeley for about an hour, and Valerie and I will be taking over the commentary for this period.
Hi everyone.
So yeah. So currently we are at the depth around almost 800 meters, and today we're really seeing a lot of creatures. The underwater temperature right now is about 6 degrees.
Yeah, it's really interesting because you can see that the habitat is completely different to the previous dives we've been doing. So yeah, we see a lot more life. The last one we did was more of a big mound in the middle, whereas right now we're more at the edge, which gives us a completely different habitat and loads of different things. The, you can even see the rubble here is completely different, so we're excited to see what we get to see today. Yeah. This looks like. I see Austin is asking about the drone work. We're both doing drone work actually, except
Yeah, we're focusing on a bit different topic.
yeah. So
Yeah.
I'm focusing a little bit more on mapping out through spectral and mixing the substrata. So I'm looking at corals, algae, and sand, whereas you're doing more sea cucumbers.
I'm
right?
doing more sea cucumber related, but still it's from drone images, and I'm trying to detect the sea cucumbers from drone images. But right now we're zoomed into look like a black coral
black coral?
colony.
It's quite similar to the last one that we collected. Very fleshy, different color.
Gonna look closer at it.
Let's see if Jeremy has anything to talk about this.
A little bit closer to it.
Yeah, if we could zoom in a bit, maybe on the polyps.
Yeah, I think definitely on some of the branches it's been chewed on or somehow being disturbed by some other creatures. You can see it's just like a few tissues are gone and just the skeleton remained that is covered by some other sediments.
Yeah, it seems a lot of people are quite intrigued by this coral, and I have to say the same. I haven't really seen anything quite like it. It's hard to really distinguish what the polyps are, to really look at the structure, and even to wonder what has caused the loss in tissue at the very tips of some of them. It's sea stars, yeah, someone mentioned. That could be a very good reason for that. But yeah, if we look closely, it really does look like it has a similar structure to the previous clear coral that we collected in the first quiver.
Yeah, I think the only difference that I can see from now is from the main stem. The previous one that we collected only have branches on either side of the main stem, but this one has longer branches on the side, but towards the front side of the stem, and has this tiny branches sticking out. So it might be another species.
Someone mentioned snails as well.
Yeah.
I think it ranges from invertebrates, and I've seen lots of mollusks can eat them. So it really could be anything. We just, can only speculate what's down here, right?
Do you want
Move
it?
on.
Yeah. We can move on.
No. I think we'll be okay. I don't know. Should we take it into the bio box? Or?
I think we have something similar. Did we?
it wasn't the same color, but it was.
Yeah. Is this
Sure,
the
try.
missing black coral
Yeah.
in
Biobox, no.
in the bio box?
I think, yeah, we'll take it and put it in the bio box.
Okay. Do you have a preference which one?
One with nothing in it, so I don't know, B, 1B or 1C. Is that possible? Yeah, 1B. Just says Benthic species. Exactly, it collected just to be sure, and we're never sure here, right? So, yeah, unfortunately, we filled both of our quivers for the genome sampling today because we just got really excited, saw very two intriguing specimens in the beginning. Usually, we would want a few more quivers so that we could, get as many samples as we can. This one has a nice critter on it as well. Which would have been good for the genome sampling, but we did fill those, so yeah, BioBox is good. Thank you.
This one is not good, so I can load them from here. So are we all strangers? We've got an interesting question coming from YouTube. Are we all strangers? Or I think a lot of people that are talking and chatting in the streaming right now, a few of them met each other before doing the expedition, but also quite a lot of people just follow Schmidt Ocean Institute and got interested in what is happening in the deep water there. Because it is true that you don't get to see this view very often in your daily life. And we pretty much do a dive every 2 to 3 days, depends on the weather and then the map that we've got to the seafloor.
Exactly, and even though we're all strangers, as someone said, we're all friends now. I think the marine community is quite a relatively small one, so it's really, degrees of separation here, and I think everyone knows someone who knows someone, so. Yeah, same to about me and Joan. We both actually study the same thing, both at James Cook University, but we had never actually met before we met about a week before this trip occurred. It is partially also because of COVID, because, we weren't really going to university, so we had met on Zoom a couple times.
Exactly.
But it was just funny because we know so many of the same people, but we had actually never met in person. So, small world.
Yeah. And Jeremy was saying the black coral is either heteropathous or hexapathous. So could you let us know a little bit more, maybe, if the heteropathous and hexapathous are the difference occurs in the way that how the branches are organized on the main stem, or they have some other characteristics that we can tell apart from?
More sea cucumbers. Just love to see them swim away in their funny little, I don't even know if I can call it a dance, their wiggle. Yeah, Joan, you want to tell us about some sea cucumbers? I know that it's not actually really your specialty. I know that Rob kind of threw you in the deep end a little bit. But...
Yeah, like I do like sea cucumbers, and my topic, this small project topic is about detecting sea cucumbers, but it's more like detecting sea cucumber as a general family, the holothurians, in the reef flat in shallow water, but not really into the species level, so I'm not very experienced with sea cucumber taxonomy. But it is interesting to see how sea cucumbers are different in depth compared to how they look like in the shallow water. And in this dive, we can see quite a couple of sea cucumber like we're seeing right now. They are sandy, pale color with, you can see a lot of sediments on top of them. And this is relatively common in my experience of how sea cucumber looks like in the shallow as well, because they usually have mucuses secreted on their body, so they can stick to the sediments in order to camouflage them very well at the sandy bottom to avoid predators.
Okay, so I've got a question. So you're obviously doing a classification to try to detect them. You said that they obviously have very good camouflage, but how do you actually detect them? Like, which
Yeah.
algorithms are you using? Are you doing object-based classification or?
Yeah, that's a very good question. I think thanks to all the computer science fellas, they really worked hard on the algorithm and different models right now, and there are mainly two categories of detection: supervised and unsupervised. And when you're doing supervised detection, which is the one that I'm doing, you can choose either by feeding them vectors or features, like you've said, to build up a model from scratch, or use another method, which is the way I'm doing, is using deep learning, which is kind of like a black box. And you just put the ground truth images to the model and train the model with the convolutionary network in that model. They somehow can teach themselves to detect the sea cucumbers. They might grab features like, if the color looks similar, but they still have the shape or the contour that is quite obvious on sand. And of course, for human eyes, we can detect it. And now it is also possible for the computer to extract all those features and then detect the sea cucumbers from the images.
Okay, that's very interesting. I'm guessing that you must be one of the first people to be doing such a study on sea cucumbers.
Yeah, it was very interesting to do the lit review. I'm not the first people to try this method on sea cucumber. There were similar approaches before, but most of the time people were doing that in an aqua form. So there was no one trying to do a similar detection from drone images or in the wild. It's more likely to be a big sea cucumber farm, and then they have divers to go inside with cameras and took a video clip and see what is inside and try to make decisions whether they can farm that sea cucumber or not.
Okay, well, let's have a look at this polychaete worm. It's a bit shy today.
It's really colorful.
Yeah, might actually, yeah, be nice to have a closer look at it, because you don't really see them from up close very often.
So the spatial resolution, coming back to the question Austin asked, it was drone images from 20 meter above. So for each pixel, it's about 0.2 meter in the field. So it was actually, 0.02. So it was like 0.02 to 0.05. So basically 2 to 5 centimeter. It was quite high compared to the satellite images resolution that we can get.
So are you looking at multispectral imagery then? Or is
No,
it RGB?
I'm just looking at RGB for now.
I'm guessing the reason is because with an aquatic habitat, usually the wavelengths beyond the visible spectrum aren't always, I mean, they sometimes can be useful, but not.
Yeah.
Because of the attenuation of the water, usually the visible spectrum is the range which we have to work in.
Yeah. What is the crab?
Just see if you'll
See a crab?
Yeah, there was a crab too.
see if we can see his little head. Yeah. We've got Vicky here with us, who's
there we go. He's sticking his head out. He's got all his antennae and palps out, having a look around. So I did a lot of work on polychaetes. on the other side of the Queensland coast here, over on the western side of Cape York, in the Gulf of Carpentaria. And there's a surprising number of polychaetes that live within the mud. This guy doesn't. He's what we call epibenthic, so he lives on top of, and he's made this casing out of all of the shells and just bits and pieces that they find. They make themselves this little home to live in, and they carry that around. And he just pokes his head out enough that he can burrow into the sand and find food, scavenge around, see what he can eat. And yeah, he's actually he's not that shy today.
Yeah. He's putting on a show for us.
Yeah, he's quite happy to come out and see what's going on.
I guess at this depth he probably hasn't seen many machines quite like our SuBastian, so...
Yeah.
You want to sample that or?
No, we'll put him back.
Just leave him to rest.
The funny thing is that we're looking at, you know, a fairly sparse surface of the sand, but underneath you can see all of these holes. So the epibenthic, the on top of the sand stuff, there isn't all that much there. I mean, there's more than what we've seen in previous dives, but there's not huge amounts. But in just a very, very small, if we took a core, say 10 centimeters diameter, and we counted the organisms within the top 5 centimeters of that core right here, we would find ridiculous amounts of life, from what we call myofauna, which is bacteria, through to diatom kind of size, so microscopic. Then we've got microfauna, which are up to about 0.5 millimeter in size, and then a macrofauna, which are above 0.5 millimeter. And within that there would just be copious amounts of life. It'd probably take me 2 or 3 days just to count the organisms that are within that piece of mud right there.
So all these shells that we're zooming in right now, are they like remaining polychaete shells, or?
No, those look like spines, maybe echinoderm spines. But yeah, that's the kind of thing that they do use though to build their homes, build their tubes. There's a lot that they build tubes within the sediment as well.
Yeah.
And they make their own little climate within the sediment, so they regulate their own temperature, their salinity within the sediment. So what's going on in the water column outside of the sediment or above it is very different to what's happening within the sediment. They look after their own little climate in there. Yeah. So while you guys are looking at the top of the sediment and what's growing out of it, I'm thinking about all of the stuff that's in the sediment.
The fascinating things.
They're just not easy to, well, they're easy to sample, but it's a very messy job.
Yeah, we see there really is so much in there. There's
So
all
much, loads. There's a
all the
polychaete
shells.
tube at the top there. See how he's taken those spines and attached it to his tube, and all of that stuff means that their tube, it doesn't, with currents or anything that might come past, they can't. It doesn't get dragged out of the sand. It gives it some friction to hold it in there.
Right.
There's some more there. There's different types.
Yep.
So this would be really interesting. That's not what we're here to study, but for me this would be really interesting to get it under a microscope and have a look at exactly how much life is there.
Yeah, all the sediment lovers are going crazy.
Yeah,
right
yeah.
now.
I'm one of those. There'll be loads of foraminifera in there. There's a gastropod over in there. Probably bivalves, gastropods, foraminifera, copepods.
Pteropods, will they be in sediment as well?
pteropods, I guess that have...
anything that's below the centimetre. Yeah, what is that? Orange bit
This.
down there. Nice little gastropod. Oh, there he goes. He's sitting on it now. Yeah, see, there's a few gastropods in there. Yeah, I don't know what that orange thing is.
I don't know either.
We have a suggestion on YouTube saying some micro mollusks.
Yeah. Yeah, there'll be tons of micromollusks in here. And they're so delicate.
Yeah, we had a sample of sediment that we were looking at the other day. I don't know if anyone watched the last ROV dive, but we tried to sample this, I think, coral that was retractable. So we tried to push core into it and grab it, but we were digging in it and there were so many little bits and pieces in there, but we unfortunately didn't actually get that coral for all of you guys who were wondering. So we had to throw that away.
Are they the sparklers?
No, that monitor is just all over.
Oh. I was gonna say that's quite different.
Yeah, the sea pen that kept hiding. I know, what a pain. Would have been really interesting to actually get to see what it was, but it somehow managed to escape. We don't even really know where, because when we put the push core in it,
That might be a
we
sonar.
couldn't even find anything.
Oh,
Is that
that's a pretty.
You ever seen a feather star swan?
Oh no.
Oh, good.
It's magical.
It's a little
Actually, I
fish.
feel like I have. It's kind of like a.
Is that a fish under a crinoid or sea star? It's yay. Yeah, there's a crinoid swimming.
It's like it's climbing a ladder.
Yeah.
Cool is that? Yeah, wow.
Big ones.
It really is quite beautiful, isn't it?
Some of them keep going pretty fast.
The booping arm always works out. Always boop. It's freezing for us. It's a bit photogenic today.
Yeah, look at the fine tip of its feet underneath.
Should just take him along on our journey with us.
I'm pretty confident it might just swim upwards and attach to Sebastian and just come with us all the way.
We'll leave you alone, buddy.
Okay. Leave him to swim off for a bit and we can continue, I guess. Because yeah, there's just been so much we haven't actually moved as much as we thought we would be. So I guess better get a move on so we can hopefully find more interesting things. We made 90 meters. Oh, very far. Another hour, we might make it another 90 meters.
Yes.
I see another. Is that a shrimp there?
Yeah, that's a shrimp. And its antennas are stretching out, and then a brittle star not so far away, trying to crawl away.
Maybe doesn't want the boop from the arm.
No.
Saw what we did to the last guy. It's a sponge right there. Yeah, that looks like a small sponge. There's actually one sponge that we've been looking for actually, that that Jeremy and I think Merrick as well is very interested in. And we've seen a picture of it, but it really looks like it's only about 1 centimeter
It's really
long. It's really, really small, so we've been trying to look out for it, but it's just so hard when it's that small and it's transparent and
It
I
looks
guess-
like a little tiny dandelion.
yeah, dandelion.
Dandelion.
Yeah.
Yeah.
And I don't think either of us are sponge experts, but oh,
Ooh,
oh wow!
look at
this
this
guy.
fish.
There's little small feet in the back. So it's a
Yeah.
goosefish.
I've heard that name before.
I think it's a goosefish.
It's a type of anglerfish. That's the little lure right in front of its eyes.
Okay, it's got. Yeah.
It's definitely enjoying its life at the seafloor right now. Not in a hurry.
No, it's just surprised to see something quite so large as Sebastian. We're saying it could be a toadfish, frogfish. It doesn't look like the frogfish that I see, I know, in shallow waters, but
It's like that.
it looks like it does kind of look like a frog. A very shocked frog. Oh, wow. Yeah. Yeah, sea toad. So, Conicidae, we believe. I don't know. Do we have any fish experts here? It is.
Yeah, it is also called coffin fish.
So
Yeah. Do we know if it's an ambush predator, or I'm guessing if it's anything like the frogfish from the shallower waters? I know that the frogfish are ambush predators, but does anyone know about this nice little guy here? Yes, I think people are saying it probably is. Yeah, I'll leave you alone.
Yeah, as you've said, and I just had a quick brush up on Google. So these sea toads are mostly sedentary fish and rely on more opportunistic way of hunting. So I'm guessing basically they just wait there and see what come past.
Yeah, and then ambush, I guess.
Yes.
Smart way of doing it, I guess. You don't have to waste your time. Is that a brachiopod or a
It's a
gastropod?
gastropod.
Leaving a trail. Oh wow. Yeah. Yeah, it's really nice to finally see, because we've seen so many trails all around, and it's always sparks my curiosity when I see the trail, but without seeing what the
Exactly.
animal might be. So it's actually good to see for once what the trail belongs to.
It's usually when the trail is there, it couldn't last that long. It's all soft sediments, and if there's any type of current, it'll just bury the trail of the from the gastropod. So they shouldn't really be that far away. But this is the first one that we've seen.
More shrimp. Oh. Shrimp trying to swim away.
But he looks like he's on some kind of treadmill. He couldn't really go further.
Exactly.
That's what the current is.
Yeah.
So how strong is the current right now?
That shrimp, that's how hard that shrimp is trying to stay still.
Yeah. The current must be quite strong.
For Sebastian, maybe for the kind of shrimp.
Yeah. Sebastian doesn't have to run quite like that shrimp has to run. Oh, another sea cucumber. So in your project, Joan, when you're looking at sea cucumbers, you said there are two different types of species that you are looking at mainly, or?
There were actually more than two species in the shallow reef, but there are two species that are quite easy to mix up.
Okay.
One is Holothuria atra, and then the other one is a very similar one. But the Latin name, yeah, it's just slipped through my mind. Yeah.
No worries, don't we always forget? And I know that you're going to be working on a different type of project afterwards, right?
Probably, but we're not certain yet. Because this is something really, especially with the sea cucumber and drone images, this is like a first one people are trying. So there's a lot of potential applications that you can use for this type of technology. For example, like Great Barrier Reef had monitoring for bleaching, crown of thorn outbreak, and just in general coral reef coverage, like the project that you're doing right now.
Yeah.
with satellite images or drone images to detect the benthic cover. But they've never really had a systematic monitoring methods to monitor sea cucumber or other sessile invertebrates.
Okay. So, obviously, sea cucumbers are very interesting. But what would you say makes them so important to study and to monitor and track?
Yeah, I guess other than its market value, which made a lot of researchers do sea cucumber studies before, trying to make sure what's their nutrient value and how much it benefits the aquaculture farms and economy, right? It has another very important ecological function that is called bioturbation, that is mentioned by Rob before as well. Basically, as you can see, even in this deep sea ocean, some of the sediments are having very structural, regular shape, and it is highly possible that they are the excretion of sea cucumbers. And this is how they perform their bioturbation functions, because they kind of just filter through the sand and putting the organic nutrient back into the food web. So they're back in the trophic level, and then they just filter and clean the sand and expel them out.
Okay, so I guess you could also see it as a way of looking at ecosystem health, or
Exactly.
looking at reef health, because
Yeah.
if you don't get many of them, I'm guessing you could translate that into, obviously, decreasing health or decline on the reef then.
Yeah, especially it is important to know the relative density and also the abundance of sea cucumber in the reef flat, so that you can have an estimation about how much sand has been involved in this bioturbation process. If you have a quite accurate estimation of it, then it would be easier, for example, to predict the volume of sand can be filtrated every now and then.
Okay.
Yeah.
And I see Jason in Michigan has a question. I know that you, as we said, you're not exactly a sea cucumber specialist, but says, how do some species selectively pick which pteropod shell to place on their bodies? Do you know anything about that? Do they have very high selection for that? I'm guessing anything that
I'm not sure about that. Yeah.
is anything that helps that they can maybe recognize with camouflage. You know, if we have actually any sea cucumber specialists out there, it would be great to hear more.
So in terms of your project, I'm also curious with the hyperspectrum images. What's the depth that you can analyze or obtain the results?
Yeah, so there's been various studies about the accuracy at much higher depths, obviously, because if you're not going to do any water column correction, often past 5 meters, that's where the accuracy starts to drop quite drastically. With my data, I am quite lucky to have done it same as you at Heron Island, so the conditions were actually ideal. So mine is kind of acting as a baseline study to see whether I'm not actually using hyperspectral imagery. I think that is the main novelty of my study. I'm looking at spectroscopy readings, so I'm looking at individual points. So I don't actually get an image out of it, but I get more just readings, and I can convert that into pixels, but the raster file that I would create and the spatial information wouldn't create a beautiful map as an image would. But the corals that I have been focusing on go up to 10 meters in depth. I just finished my preliminary classification, and it actually did manage to classify the corals even closer to up to 10 meters. So
It's another handsome shrimp swimming.
that is a nice one. So yeah, I know that a lot of studies do have pre-processing to do. They
Water.
correct for the water column, but because my study is kind of trying to find the easiest way, but also the most cost-efficient way to map out a coral reef. I'm actually trying firstly to see if I can do it without a water column correction to see what the results I can obtain are and if those are accurate.
Yeah, I think that is very important. I think there's also one big aspect of my topic is trying to use as little images as possible to reach the goal to detect other sea cucumbers. Otherwise, we're trying to let the computer do its job, and if it couldn't be automated with a relatively small data sets, it doesn't really reach our expectation.
Yeah, exactly. So I think coming up with the process of automating everything is the hard part, but once we have done it, then hopefully people can kind of recreate the process in a much easier way, and it'll just facilitate not only sea cucumber detection, but also just coral reef mapping, which would be incredible because in some places, in remote countries or where they don't have access to a lot of very, very expensive technology, they could just get, a consumer grade drone and, do that study themselves without having to access technology that costs thousands and thousands of dollars just to, map out their reef, to try and determine more effective conservation action, or to try to detect where they have sea cucumbers. So.
And we've got another question for you, Valerie. So
Yes.
when you refer to the water column, what does it mean exactly?
So if anyone out there dives, you obviously know that the deeper you go, you get different wavelengths that get attenuated. The deeper you go, that's where the spectra that will be recorded will be not evenly affected. So because of that, you have to try and there's many complex algorithms that you can look at, and equations where you can input, attenuation coefficients, especially when you have a water column that is quite murky. So you have to take into account what is in the water column that might cause differential light refraction. And there's plenty of different components along with depth. As I said, depth is really the main one, because that's where you start to lose colors such as red. Almost after not even a meter, you start to lose the red wavelength very quickly. So you have to account for that non-linear loss of wavelengths.
Yeah, we have a lot of
Yes,
sand filtration.
yeah, and as Austin said, for the non-remote sensing nerds, raster is essentially a pixel image. So it is, if you see a grid, usually you get a continuous length of pixels, which creates a raster. So it's almost like a visual matrix format, but with these pixels. And because my data format was not in that, and a lot of the algorithms that I needed to use to map out these coral reefs, I needed a raster format. I had to find a way to convert vectors to a raster format, which yeah, it is possible, but it just is an extra step in pre-processing.
Yeah. We also see a couple of these flatfish today.
Yeah, I think that's a sea cucumber with the camouflage.
All in the name of science.
Yeah, more sea stars.
We'll look at it because specky cucumbers are.
Is there one specific thing, Joan, that you are looking out for that you would love to see on, not specifically this dive, but any of the dives coming on the Falkor?
Yeah, I think I'll be so excited if you can see an alive Nautilus that is swimming. But these sea cucumbers are so cool as well.
Yeah.
This is the first pink orangish color sea cucumber we've seen with all the
It's
spikes.
really quite beautiful.
Yeah.
Little curls. Yeah, it is beautiful. Yeah, Nautilus would be fantastic, obviously. I know on the last trip they
Yeah,
did
where
manage to see quite a few of them.
Already did depth forms.
Alive ones?
700 meters is this sweet spot for us.
Right. So our ROV team just told me a very good news, saying we're around 750 meters right now, and when we reach around 700 meter, we do have a chance to see the Nautilus alive swimming in the water.
So we were actually just speaking about the Nautilus before this dive started, and I was actually saying that it is curious to see why people love them so much. I mean, of course they are.
The shell is gorgeous for
symbol.
sure.
But you know, they do have the status as a living fossil, and I was telling them that I feel like I had read a paper somewhere that that actually talked about how high the genetic diversity of the Nautilus was, which probably indicated that even though we believe it's a living fossil and that it's preserved its diversity for so long, I mean its genetic code for so long, the shell doesn't look like it has changed over the past hundreds and thousands or millions of years, whereas it is believed that the tissue actually has evolved a lot. And people do believe that the Nautilus actually evolves faster than humans. So
We've got a little tiny jelly swimming in the screen now.
yeah.
Is our jelly expert Dougal somewhere online?
Anemone.
Yeah, it's out of sight.
Let's go back in and see it. It's not shadow.
Yeah, some anemones in this.
That's a
Can you tell if that's a sponge next to it?
I feel like it might be a small sea cucumber. And the shrimp hiding under an oyster shell. Yeah, he get himself a very cozy home here. Yeah. Unfortunately, the fish just swam away. We couldn't see it through any cameras that we're having right now. But I'm pretty confident that we're going to see a lot of marine lives today. For all of you that has just joined, little basic information about the dive we're doing. This is Falkor research vessel with Schmidt Ocean Institute, and we're doing the cruise trip north and depth of the Great Barrier Reef. We're trying to map the outer edge of the Great Barrier Reefs. Today it's our 398 dives, and we're trying to do a long and shallow dive. We started at around 850 meters, and we're trying to go all the way up to the shallow for as much time as we can.
Yes, I think we're planning on being live until we reach about 150 meters depth. I think that was the plan. But with these dives, obviously, as we've seen previously, we can't really predict exactly what's going to happen, how long they're going to take. Depends what we find, if we have lots of sampling to do.
But the first 100 meters is definitely exciting. We already collected 2 push cores and two quivers.
And then we have
Bio
two of
box.
our bio boxes with some sponges and some black coral that we found.
And for now, the substrate type that we've seen underwater are mainly fine sediments. We haven't really seen any big rocks yet. But compared to the dives that we had before, the diversity of marine lives on the fine sediment is a lot higher. As you can see from the inset of the map and screen right now.
And that was about a week.
We have Rob and Marty who have just come in. I think they're giving their lecture at the moment.
That was collected during that leg. Leg 2 is from Cairns up to this Cape York area, and it's 3 weeks in total. We'll then head back to Cairns. Some of the science crew will disembark. A few other new people will join,
This little
and then
flounder.
leg 3 will take us out.
See, on this one we can actually see a bit more of its patterns rather than the last one that was completely covered. So on our last dive, most of the ones that we saw were like this, and you could see their beautiful patterns that differed on each of them.
accidental. Yeah, just while we're in the control room, maybe we'll just have a quick wander around and then I'll answer that question. So this is where the ROV pilots are sitting right here. We're seeing J-Rod flying the ROV just at the moment. And then just
Yeah,
on the
Joan, have you actually ever watched the Never-Ending Story?
No, I would love to, but I only heard that movie after I'm on board.
Yeah, so neither have I. And I'm guessing most of you guys know that the Falkor is actually named after a character, I believe.
Characters in the
If I'm wrong,
movie?
in
Yeah.
the Never-Ending Story.
PI chair here is Valerie and
saying
Joan,
hello.
master students from Geological University. And on the event blogger, we're
Saying hello to the students at UC Berkeley.
below from the University of Queensland. So there's quite a few people in the control room at any given
So
time.
yeah, we definitely have to make it our mission to watch this movie because we know that we have two of our side boats that are also called, that are also named after characters from the
Valerie
movie.
and Joan are talking live on YouTube, and if you have a look over here, this is the chat on Facebook and YouTube. So we have them running, and we're able to watch questions that are coming in and comments. So this is how we reach out to experts ashore. You know, we're only a small science team, so we have to rely on people ashore helping us, not only amongst the other principal investigators,
It's a
but also
echinoderm
many
shell that is
experts
coming out on the
who are
screen.
watching the live YouTube feed. So, for
Oh yeah,
instance,
that looks like
here
a
we've
dead
got,
sea
this
urchin shell,
flatfish has to be
Carnivorous.
a Seafloor. So we have fish people who can give us the names of the species of the fish as we're watching in real time. So it's a two-way
Oh, fish
feedback,
coming
and
up?
for us it's
Yes.
really
So our
educational.
fish
because
finder
we're learning
is
a lot from it.
telling
but
us
we're
that we've
also
got more
showing,
exciting things coming.
Talking to the world essentially during these live YouTube feeds. So it's pretty
So
exciting.
we're approaching to the fish from another sit camera that we're having. There's a quite relaxed fish right here.
Sorry, should we stay in control room here or should we move out?
Hi fish. So,
It's quite big eyes, this one. And it's again a rat tail.
nice rat tail.
Yeah. This rat tail has less profounding dorsal fins than the one that we saw around 1700 meters. Is this a variation within the genus?
Yeah, do we have any fish experts out there that know the genus or the species of this rat tail?
For me, it was just
I'm on.
a combination of already being a recreational
Hello, J-Rod's mother.
Yeah. Shout out to J-Rod's mother.
subjects that we enjoyed in my undergrad bachelor's degree, which was the geology, sedimentology subjects. But I wanted to combine that with marine geology. So yeah, it was just pretty much just a case of
We haven't seen any gulper eels for now.
continuing to pursue
Yeah, this is our third dive as well, so we haven't seen it in the past 3 dives, but we've seen it in gulper eels in previous dives.
as a student opportunity. Definitely seen it on that was my first time going on board research vessel. Yeah, and I've just built on those experiences since then. Resume, just a series of
Yeah, those gulper eels would be fantastic to see. Really looks like those deep sea monsters that you hear about, I guess, growing up.
Got another echinoderm coming out.
And a nice little flatfish in the back. Again, showing its colors, its patterns a bit better this time.
being in the right place at the right time and knowing the right people.
It's more than that, Marty. You're very good at what you do. It's really a pleasure to have you there.
Ooh, we have this rock coming in our sight. Usually when rocks comes in, Jeremy and us will get exciting because there will be always teeny tiny things attached on it or just hanging on one of those overhangs.
Even from here, you can already tell there's just so much on it, and you can already see the polychaete worms, I'm guessing.
Yeah, let's take a closer look.
Yes. Everyone's getting excited about the rock. Sponges, polychaetes.
ending
Oh.
up mapping.
Yes, unfortunately, Dougal, I think you missed one teeny tiny
And I
jelly.
was always left with this question: what's down there? It wasn't enough just to make lines on a map. I needed to know.
So you can
what
see
was
some
down there,
polychaete
what was the shape of
worms
it, how
to
did
the
it
left
get
there,
And so
some
that's really led
sponges.
me. I left the Navy and ended up in academia, and I finished my PhD
Sadly,
in
we can't lick the
2005,
rock.
And I've been mapping the seafloor.
I guess
ever
our
since.
version of
And
the
so
lick is the
for me,
boop
it's just this
in this
long
case.
quest to understand the shape of the Great Barrier Reef, but importantly to find what's living there. And so that for me is this huge revelation to go from this geophysical three-dimensional mapping to actually understand what it's made of, what is the rock, what's the sediment, what's living there. So it's very, very satisfying to be out here on the Falkor and bringing all of these tools together in one expedition and see what's living here in the deep Great Barrier Reef.
Yeah, I have to agree with
So we've got Joan learning to use the camera that we can actually choose what to look at. It's our first time getting to play around with this, so it's also very exciting for us.
It has a lot of encrusting sponges on top of the rock.
It's the imagination.
Yeah, and of course, we don't have the answers to everything. We're seeing pictures of the seafloor and we just go, what is this? It's kind of fun, because we're seeing it for the first time and we're trying to make sense of it.
I think that's a brittle star
We're talking long geological time
back
in many
there.
cases. Certainly in this part of Australia, and probably arrived in the tropics
I was,
around
like,
about
thinking
22
of
million
that.
years ago. So that was the first time any coral reef could have started development. And then we've had this long period of erosion since then. And we're looking at certain features that we really can't make sense of. Hopefully by the end of the expedition we will. But I guess this is the fun part of science. You get to see things that no one else has done. And for us, it's this sort of constant burning curiosity. What's down there? How did it get there? And really what it leads to is more and more questions. We don't have the answers. The fun bit is that you
Look at that shrimp.
add the questions.
Oh yeah, I didn't even notice the shrimp there.
And this is,
It didn't have the glow in
this is
its
the great
eyes
thing.
that usually
When
pop
you can
out.
bring an expedition like this together and bring students, they bring fresh eyes, new ideas, and this is how science develops. So we're very aware that what we're doing here is a legacy for the future. We will not have all the answers at all from this one trip. All the
There's a
samples were
sponge as
taken,
well.
3D maps were collected.
Yeah, there are a lot of
They're gonna live
encrusting
on
sponges.
for years and years.
Huge variety
They
of them.
use for scientific
I almost
data
wish myself
samples
I
for
knew
many people
more about
along
sponges.
the expedition.
It's funny because trips like this
whenever I go
really
out to sea,
make
it's
you
very much thinking about the
develop
future.
new interests,
Long
I guess.
after I've passed
Yeah,
from
I think
the
this is a little
Yeah,
squat
squat lobster. Yeah,
They're very hairy claws.
very long arms. Doesn't really have to leave his home burrow to catch some things.
Move on.
Oh, a
Oh,
little
there's
jelly.
a little jelly. I don't know.
Is Merrick or is Dougal
Yeah.
interested, Dougal, to make up for the lost jelly that you missed?
It's moving out of the range a little
Well, I think
bit.
that's a pretty easy question to answer. I would like people to have a better appreciation of the
I think
Great Barrier
Dougal
Reef
needs to go
beyond
back and
just
check the
coral
video
reefs.
again.
Now, coral reefs are beautiful,
Um,
very important to Australia and the world. But if you think of the Great Barrier Reef Marine Park as this huge collection of different ecosystems from the shallow
As
to
he
the
says,
deep,
I spy a
what I hope
crinoid. It
is
really is a
It's
game of I spy,
data
isn't it? There's just so much happening
will
on one
open
screen. You
people's
can
eyes
really
up
be
to
looking at
understanding
anything.
this amazing structure, the world's biggest coral reef ecosystem. That
there's so many things just happening on one rock.
what look like in the deeper water. So I hope that is the bigger legacy, that we can open people's eyes to what is really
No,
I don't think so.
I think.
Yeah, maybe
Yeah,
move
I
on
think we
to
can
the
move on.
maybe next rock.
Hopefully, we'll find another one.
Shallow coral reefs
Well,
cover
fine.
something like 7%.
Sorry.
Well, fine.
Oh,
So
okay.
most of the Great
Yeah.
Barrier Reef marine park heritage area shallow coral reefs. And all of those other areas need to be protected, need to be explored, we need to know what's there. So yeah, I think it's not just shallow coral reefs. There's so much more to it.
Yeah, and it's not just some academic exercise. It's the
We
ocean
got another
that
black
interacts
coral.
with the deep
Oh,
sea.
that looks like the one that
But
we
it-
sampled, I
Yeah.
believe.
That goes over the chips, brings cool water onto the coral
They
reef.
really
So
are quite
these
beautiful,
ecosystems
aren't
are
they?
not
They
disconnected,
do look
they're
like
all
a
connected
much
from
larger
the deeper
black
waters to the
black coral
shallow
sea
and
pen
vice versa.
almost.
So
Yeah,
understanding the shape of the
Wow.
seafloor, what's really down here is really important for the Great Barrier Reef as an ecosystem as a whole. So really, I just hope that our what we bring.
I'm still trying
from
to
the
get used to the controller of the camera.
It's opposite. It's
Great
up,
Barrier
down,
Reef
and down.
And when you use the camera, remember you're just controlling the camera.
Yeah.
ROV, so
Yeah.
you want to go, so.
Yeah, wow, that's beautiful.
Strawberry creamsicle.
Even more pristine sample than the one we collected, but it's
Yeah.
probably actually better to leave the beautiful one here.
I'll sit down so you can look at it.
Yes, definitely less nibbled, so hasn't fed as many critters around.
Yeah. Are we going up on a hill or a ridge right now?
We just did a vertical shear phase, but pretty.
Yeah, this is a much bigger rock than I first thought it was.
Jump on the YouTube chat and send your questions through.
Yeah, that's fine. Yeah, thank you, Hannah, and thanks, Bethany, and everybody watching.
That's perfect. We can have like
I mean,
See you later.
Can we find any critters in there?
auto.
Some little
There you go.
nursing.
wow.
Oh, wow. It's a great shot.
I remember previously we've got a question saying how do you count the 6 tentacles in the black coral? Because they do look like paralleled pair tentacles all the way down the branch. But I think from this one we can see quite clearly the darker, 8 tentacles. I think 6. Octocoral software. Yeah.
Yeah, just count.
Yeah. But I think this one has a very obvious central mouth kind of mouth tube that you can see in the middle. It's darker than the other tentacles that surrounded it. So there are 8 tentacles. I can't really count.
Yeah, stunning indeed.
But yeah. I think we can.
Yeah, probably move on to the next thing.
Don't like Chuck. More right here to the right.
So there's that rock. Yep, we found the rock.
Yep.
Yeah, I think Marty and Rob are finished with their lecture. So
And it's right back on time for some exciting rocks.
Timely moment.
There are a couple of big rocks here.
Okay,
Yeah.
let's go take a close look.
We haven't really poked onto it to see how hard it is. We're just mainly checking the marine lives on the surface.
Okay, good.
Yes.
Is that a sponge there?
That orange, I think it looks like. Oh, yeah. I'm not sure.
Anemone. I'll sit down in front of this one and get a close look at it right there. I'll sit on top of this rock, look at the other rock.
There's another, I think, rat tail is on top of this rock.
Do you want to look at this rock?
Yeah, we could get closer to the rock and have a look at it.
Covered in mud, so it's going to get muddy for a second.
Okay.
Ooh, jelly!
There's plenty on this. If you zoom in into some
Yeah.
of these.
Yeah, I'm controlling. I forgot.
You've got the power drone with the controller.
Yeah.
Hold on, I'll get
Yeah.
right down there.
Okay. Yeah. No worries.
Yeah, I just have to wait till it clears up a little bit before.
I can sit that down. Forward.
Got a question on sea urchins. Like, we've seen quite a bit of urchin tests, but we didn't really see many live urchins. I might be wrong, but somehow I have this memory saying urchins are a bit nocturnal, so during the daytime they might hide themselves under the crevices or underneath the rocks, so it's not that easy to spot them.
I know during the last dive we did see quite a few. I don't know if this is just a habitat where we don't see as many. We have seen, yeah, as we said, a few of them and a few dead shells from them, but I don't know, Marty, do you have any theories why there may be more dead urchins at this site?
No, is the short answer. No, I've been distracted for the past hour or so, so I'm not really up to speed with what you've been seeing.
Yeah, we've seen a few live urchins, but
Mainly just the
we've
shells.
seen quite a few shells,
Okay.
and really not even close to as many as we've seen the last dive, because I know we saw
Yeah.
like
Quite a
loads
few.
and loads, whereas here we've seen, you know, one here and there, but it might
Yeah.
just be the habitat is, you know, not.
Yeah, I
it's
mean, we're
not
in a
somewhere where
different.
they've settled. Yeah, it's completely
Yeah,
different.
we're in a different sort of setting to what we were on the last dive a couple of days ago. So yeah, different, I guess, oceanographic conditions and different. Well, what are we here? We're at 730 meters. I think. Yeah, the dive the other day was a bit shallower than what we are here.
And I guess this one is more on the edge.
Yeah.
You've got lots of tube worms here. Yeah. It's really
Is this possibly be a sponge?
which one?
This one.
Yeah, I'm not quite sure what this is.
Touch it.
Yeah, do we want to boop it? Does anyone out here have any ideas? Yep, a sponge? Okay, so it is a sponge. Probably best to wait a bit till it clears up. It's still a bit hard to see. Spicules.
I understand it from that way, but I want to see what. Take your camera for a second. That's where it's coming from. Huh? Alright, I shoot through it for a second.
spicules covered in marine snow. Yes, indeed. Lots of marine snow down here. It actually, it's something that when you think about marine snow, I had studied it quite a lot, obviously during my undergrad or my master's coursework. And I always wondered why it was called marine snow, but it was not until, I guess, these past few dives that you see all these gigantic rock formations and this marine snow that is deposited on all of them. It really does look like snow, and it's quite a fantastic sight to see these, as I said, these huge formations that are covered, and it almost looks like, you know, the tip of mountains where it snowed on. And I guess I'm more of a shallow reef diver. It's really not something that you tend to think about.
That often. No.
You see a squat lobster up there hiding.
Yeah.
Yeah.
It's a little cup coral skeleton.
Little fish there as well. Yeah, we've been seeing quite a lot of fish today. Kind of different.
I think that's just a skeleton left.
Nice cup coral.
Scrape it. Go poop it.
So we want to scrape the rocks.
Yeah.
Are we sampling
You'll be
or?
able to grab a bit from there. There's no sticky out
Gonna
bits.
steal your camera for a second, Joe.
Yeah.
So this looks like it could be a boulder, a reef block that has come down from higher up above and come down the slope and settled here. I don't think it's an in situ outcrop. But if we can have a good look at it and give it a bit of a scrape, it'll tell us what, if this is a reefal limestone. It looks very weathered, very bio-eroded, which tells me that it's pretty old.
That's hard.
Hard rock. Can you get a clean, scrape a bit of a clean face and zoom right in on it for us, please? Because I don't think you'll be able to sample any of them too hard.
Sorry. They're like knockoff M&Ms maybe? I don't know.
Chocolate.
Smarties.
Never had one, so
The Smarties?
Yeah, we've got
This is like M&M chocolate.
Smarties. I think those are smart. Yeah, we've got snacks going around the control room.
Because it's lunchtime
Yeah.
out here.
alright, I'll give a push with it.
Yeah, I think I would agree a bunch of tentacles would be those
Acorn worms.
worms. Yeah.
Alright, I don't, hold on, I'll fly it back up.
Yeah, no, you're good.
Okay.
Hand up the move my arm.
Now Marty wants to have a good look of this rock.
Because superficially it looks very similar to the rocks we collected a couple of days ago off Tideman Knoll. But this is deeper, and I would say that this reef block has fallen down and settled here. Is that as close as we can zoom?
Alright, you can head back, camera back now.
Can you zoom right in, please?
Do you want towards that angle or towards the front?
The bit that was just scraped off, the bit that was just scraped clean. You can just zoom in on that. Yeah. Yep, so this, yeah, for sure, it looks very similar to what we saw the other day. It's a reefable limestone, but probably very, very old. Lots of weathering and lots of bioerosion in there. So So it's going to be really hard for us to tell how old these rocks are. But that's all good information, so thank you.
And we've got a amphipod crawling.
Yeah. Marty, can you tell by I guess the manganese crust, you know, how recent that was deposited, or can you tell that by probably not just looking at it, but
Yeah, so that's a really good question. So these rocks are all coated in sort of either a ferromanganese oxide crust or an iron oxide crust that those minerals precipitate out of the seawater sometime much later after the reef rock was formed. And we don't really know the timing of how long that process takes or how quickly it happens. So a thick crust, if there's a thick crust, we don't really know like how many years worth of precipitation of minerals is that. But what we do with these rock samples is take them back to our lab on shore and we cut them into thin sections, which is like cut a little small slab of the rock and you mount it on a microscope slide and then you grind it down until it's only 30 microns thick, and then you can examine it under a petrographic microscope or a light microscope. And then they will also do geochemical analysis, trace element analysis on them as well.
oh, yeah. So I guess all these rock samples that we're collecting really can tell us so much about, I guess, because we know so little about this deposit.
Yeah, definitely.
It will probably help
Especially
us.
once we get sort of quite a lot shallower, up around the 130 metres. We know that in more recent geological times, but I'm still talking, 120,000 years ago, but in geological timescales, that's pretty recent. There were various sea level oscillations, so periods of time when sea level was close to what it is today, or a lot lower, about 120 metres lower than today, and that coincides with cycles of glaciation, glacials and interglacial cycles, where you get your relative sea level rising and falling, and that creates, you get new reef growth, or you get a hiatus where there's no reef growth and you get erosion, and that can create sort of terraces on the reef edge, and so that's where we like to collect our rocks from, because those are a bit younger and we can date those.
So kind of similar to the last dive that we did in those terraces, I'm guessing, that we got to see, or?
Yeah, more younger than that though, more recent
Okay.
than that. That was still pretty ancient, like I'm talking millions of years versus hundreds of thousands of years. Yeah.
Where do you want that?
The cup coral? If you can get it, yeah.
Take your camera again.
Gonna
Where is
sample this cup coral from Artie.
it? Over there. Can you reach it?
I don't know, maybe.
Move where? Alright, can you move?
I can get my wrapper off my lollipops, that's my main problem at the moment.
Jason is asking if you flipped over this big rock, would the bottom have manganese crust too? I'm guessing it depends where the rock is from. If it hasn't been exposed to
In the
the oxygen in
water.
the water column, then probably not. I guess this has fallen from somewhere though, so it could have been exposed beforehand to the water column and the oxygen, and would have had this manganese crust on it, but I guess it wouldn't be as thick as the side that we are looking at right now.
Good there, Christopher.
Alright.
So I've got a question.
10 minutes ago. I don't know if you know where. Has it gone past it?
10 minutes ago.
Yeah, I'm sure. It's
It
right.
could have been a little bit before.
There you go. There you go.
Got it?
Look at that grab.
Wow, nice. That precision.
Good job, buddy. Where you gonna put it, Marty?
can go up 1A with my other stuff as well.
So 1A is the Marty tour. Alright. Going to head there for a second.
Yeah.
Alright. With that's rocking, right? Right. Okay. Push it out.
You get that full count. That porch.
So I guess I'm guessing me and Joan will probably be heading off to lunch once Rob comes back to take over. But I guess one last question I wanted to ask you, Joan. Obviously, we've been having an amazing experience on this boat.
Yeah.
Is there one thing that has been your highlight so far, or anything that?
Your birthday cake.
Hey.
Oh yeah, it was Joan's birthday yesterday, actually. So
Yeah.
That's it, ROV.
One
Apparently-
day, right?
Yes,
Marty?
thanks. Yep.
Hold on.
Yeah. Cup Coral is back in.
Yeah.
Thank you for the birthday wishes. So this is apparently my first birthday on such a big vessel, and the chef here, they're really amazing. They baked the biggest carrot cake I've ever seen, with very nice frostings on top.
I think about
yeah, it really was a delicious cake. I guess that's one thing that we're spoiled on the boat because we really get. The chefs are amazing, so we've been eating very, very well. And I really can't complain. I can't say that I eat much
No.
better at home.
We ate much regular and had a lot more diversity compared when I'm cooking at home.
Yeah, it's really been great.
Alright, now do you want to keep looking at the
So
rock, or are we done?
can I just yeah. There might be a tiny sponge while we were looking at. I'm not sure if Merrick's still there.
How's he moving? Is it Eric?
I think it may have been on the previous rock. That's unfortunate.
Because when I remember he was asking, we were talking
this
about
one?
this,
Oh, yeah.
and then we go up a little bit, right?
It might be up.
Oops.
We get you a little closer if we need to.
This one. Does this too look like sponges to you?
So it was a small white sponge with a fruit edge. Okay. Yeah, it says we're back here again in the right place.
Okay.
Yeah.
Just have to look around, I guess.
Yeah. So we're moving slowly so Merrick can have a chance to talk to us. And is there anything here you'd like to sample? These are glass sponges and rock lithoted sponges. This is actually my first time seeing glass sponge when I joined Falkor for the ROV dive. And when we were sampling them in the previous dive, I was really surprised how glassy they get.
Yeah, if you have them between your fingers, they really do have a little bit of a crunch to them.
The spicules are made of silica.
Yeah, so the spicules, as Marty said, are made of silica, so that's why they're really so fine, but they can also just almost dissolve between your fingers, like if you had like glass.
The visual strands that you'll find them look exactly like the glass that we use for fiber optics.
Oh, okay. So I guess that, yeah, I know that Whitehaven Beach is made of 100% silica, and I know that for NASA they used that silica to build their lens.
Okay.
So I think it's the only time they've ever let people use the sand at Whitehaven Beach in the Whitsundays, if people don't know. And it was to build these lenses for NASA, and normally it's actually illegal to take the sand because it's 100% silica, which I don't think you really find anywhere else in the world.
Yeah. Yeah. The sponges are all good here.
You guys can jump out and have lunch.
Anyway, Joan and I are going for lunch, so thank you for having us and letting us speak so much.
Enjoy.
Feel free. Alright. What was for lunch today? What did I miss?
Build your own sandwich.
Build your own sandwich? Alright, let's keep going. Just keep going. If you do happen to see any rocky bits that look like you've got a sticky outy bit that you could grab, we'll do that. Yeah, but if... Oh, hang on, what's that there? Is that a sponge? Get in and have a closer look here. I think it's a sponge. Looks like a glass sponge with something entangled in with it. No, I haven't. That's okay. I'll get something after.
I'll go
Yeah.
grab some-
All right.
something to eat.
Wee. Hello. Stand by. Okay. All right. I just need to step out momentarily. I will leave you in the capable hands of anyone that wants to talk to you.
So are we passing on said sponge?
Don't need to collect that sponge.
Alright. Find you a nice rock. Oh, that's awesome.
Thank you.
Yeah, don't need to collect it, says.
Maybe you want to take the mic?
Sure. I thought these guys were going to. Oh, a bunch of smarties. Okay. Well, it's Vicky on for a little bit until everybody comes back from lunch. Where I think the guys are eating Smarties and lollipops for lunch today. Little fish in there. Trying to hide from the ROV. Yeah, it does blend in quite well. Where's our coral? Down the bottom there. And a sea star. So this is all very different kind of life to what we were seeing earlier in the dive when we've got all soft sediment. And generally on soft sediment we've got our epibenthic stuff, but most of the life in soft sediment lives beneath it. Whereas when we've got the rocks, there are a few burrowers on rocks, but generally they'll all cement themselves to the surface and And so we've got completely different kinds of communities that live on the rock surface. And it's not dissimilar to what you see in like an intertidal zone, where you've got your rock platforms, and you've got water coming and going. Of course, it's different species. The species here wouldn't survive in an intertidal zone, but similar kinds of communities where they all cement themselves to hard surfaces and don't really move from them. Oh, big shrimp.
Jeremy says grab it. Jeremy said grab it. Says on the chat, grab it. You see that picture?
Jeremy said grab it.
Jeremy said grab it.
Was it?
Okay, yeah. No, I can't see that chat. Sorry, Jeremy. Was it Jeremy or Merrick saying they want it? Nice. Ah, grab it. Ah. You can tell I usually sit in front of the data entry computer instead of the microphone. I'm not even looking at the chat, sorry guys. So we'll take this black coral for Jeremy. And I'm assuming it's in the bio boxes. Recording. Do you want the whole thing, Jeremy, or just a part of it? Can you zoom in to see what it might be first? There we go. And Merrick's also wanting to look at some glass sponges with blue tubes to the right as well of this one.
She's got it over here. There.
Those ones there. There we go. Beautiful. Right, and we got Marty back.
Okay, you're welcome to stay here.
No, you're all right. I'm good at data collection, you're good at microphones.
Alright. Where are we at? Just looking at the sponge?
Jeremy wants this coral.
Yep.
And Merrick wants to look at the sponge.
Okay.
So
So we're just looking at the sponge for now, and then we'll
we
look at the
asked
coral.
Jeremy if he wanted the full thing. I don't know if we got that answer yet.
Let's check and see. Okay. Jeremy, is that view good for you? And can you let us know if this was something that you were wanting to collect, please?
I love these reefs. Freaking useful. Yep.
Can you look at it?
Can you look at it inside to see how it branches?
Do we send that camera out?
Just give us a moment here, Jeremy. We'll just give you another view from a different camera.
Should I mess with them? Gonna go negative? Go negative.
That makes it stand out. That looks
That's the
amazing.
negative image of it. It's a little bit more of a strut shot.
Yeah, I see. Okay, Jeremy says that that is very interesting. It looks like it's a new species target that we didn't collect on the last trip. So I guess this is the one that he's been looking out for. Okay. So Jeremy, would you like us to break off a fragment to put in the quiver and then take the rest of the colony into a bio box? Bio box. Okay, whole thing, whole colony into the bio box. Okay.
So right here, looks like the main base is right there. Obviously, this one's zoomed in and that one's zoomed out.
No problem, Jeremy. That's a pleasure. So Jeremy, just to clarify, you were saying after the last trip that there was one group, I'm not sure if it was at the species level or genus or family, you said there was one group that we didn't see. Is this the one?
Where's this going? In C.
Will it fit? Or do you want to put it in the back?
It fit better in the back.
Yeah, we'll put it in the back. Thanks.
First time I just saw it once. I bumped it. Now I'm going to.
You're good then, look. Oh. Just let zoom in and let your card line up.
Alright, just opening up 4. 3, 4.
So it was it was a whole family that we didn't collect anything from on the last trip, but this is not part of that family. This is another species. Jeremy's saying this looks like a very weird colony. Do you mean the branching pattern is unusual? Okay, well, we'll get this one into the lab and take photos. we go. Are you enjoying the Smarties?
Hmm?
Do you like the Smarties?
Is that what they are?
Undecided?
Like they're wannabe M&M's.
Yeah.
Posters. Posters.
There we go.
What was Merrick's call on sex space?
He's just, he hasn't indicated that it's something that he would like to collect.
Okay.
So I guess we will carry on.
Let's go racing, boys.
Shake and bake.
Drag them back. A little bit of shake. A little bit of bake.
I'll get it.
Not first class.
What about it?
Come on, Jack. Come on, dude.
Okay, sorry, an update from Merrick. If that is easy to grab, he would like to take it, please.
Skirt. Will that eat break?
Oh, back we go.
Like that.
So we'll just get back in there. Yeah, that's no problem. There is a little bit of a lag, a little bit of a delay between what we're seeing here in the control room versus what you see on YouTube. So that's why we sometimes do a little bit of toing and froing. Do we want this? Do we not want it? Are we moving on? Are we staying? We've got our colleagues onshore that aren't on the ship with us, but these collections are really important for a whole bunch of other scientists, so we just need to be patient and get the right things for the right people. Wow. I don't, is it?
Yeah. They grinded the.
Yeah. It's like a mirror. That's in there.
Is it under a little overhang? Is it in a little crevice? Yeah, it's not going to be. No, we'll keep our eyes open for another one. If we see another one during the dive, which is a bit easier to collect, we'll put the brakes on and grab one.
Good try, Merrick.
All right.
No, what they say?
Smarties came first, M&M's came second.
Yeah, they did in
Just
my
'cause
they
the
did in
first
my
one-
world.
just 'cause
Smarties
you're
came
the
first
first
in my
doesn't
world.
mean you're the best.
Actually, in New Zealand they're called pebbles. So they're pebbles in New Zealand, Smarties in Australia.
So Smarties are chocolate. Chocolate.
Candy coated chocolate.
Okay, so Smarties in the States is like this chalk candy. That's like flavored chalk.
Chalk.
It's not chalk. It's that.
It's what it
chalky
feels
texture.
like. Right.
Yeah. So it's like fruity.
Okay.
It's strawberry.
A little sour.
Yeah, sour. Sometimes bitter.
Mm.
Like a Pez dispenser.
Uh-huh. Uh-huh.
Yeah, those are Smarties.
And this,
This is one of these urchins that we saw the other day on the previous dive
and this is an
with their little white snowshoes on. We were talking about
little snowball.
snowshoes. Snowshoes. Yeah.
You know, little Debbie snowballs.
No, they don't.
No.
Little pink things with a coconut all over the top.
I don't know what we had those. I don't know what they're called though.
They're delicious. I like the butter butters.
Another one.
Okay.
Zebra cake.
Keep your eye peeled for any bit of rock that is sticking out that would be sampleable. What is that? Is that a zoanthid colony? What, you just jumped on? That's right. Mhm.
Looks like a crab. Looks like a hobo crab.
It is.
Oh, it is too. Hobo crab with a zoanthid colony.
It's like the big brother of the little guy we seen earlier.
It's like a lion. Male lion. His mane.
Yeah, I don't know. This is getting controversial on the YouTube chat now. They're saying Skittles are better, are the best.
Skittles are off the chain.
I cannot agree.
If we're going somewhere else, I mean, Sour Patch Kids.
Shake and bake.
Sour Patch Kids.
Bring about me and Skittles.
Yeah.
Mix the two together.
Sorry, Ray, there's no votes in here for Skittles. All right. So this is another one of these hermit crabs that has... So I assume that the hermit crab is in a shell and the zoanthid colony is living on the shell.
Some that are just a crab.
Without...
Like a regular crab.
Oh, okay. Not a hermit with a
They actually
shell.
like kind of hold on to it. You can see
Okay.
the legs usually. Then you have the hermit crab version.
Should we check out those? Carpetbagger crab.
They can get 2 pounds of it. They can cook whatever they want.
Oh no, those other ones.
Sour. You ever had the ice cream?
So there's another whole other chat going on our science team's Slack channel where everyone is giving their opinions about Skittles, and I'm afraid that I can't share it with you.
It's fun to get Australians on Kit Kats. They get all bent out of
Okay.
shape about Kit Kats.
All right. I was just wanted to momentarily look at these sponges just to see if that...
Gotcha.
Is that a little jelly that's sitting there?
That little
That's-
Medusa?
Yeah, that little Medusa. It's just sitting there resting on the rock.
Yep.
Oh,
It was.
there we go. Wow. It's a little cutie. I bet that is tiny.
What is?
Wow. Yeah.
It's like
Our Dougal's saying sample, please.
You want a sample?
Dougal would like to sample that if you can, please.
It's gonna be a little tough
That's
right
going
now.
to be tricky.
No, don't be hard. It'll be alright. I'm just gonna porch out there.
Just keep an eye on it.
The laser beams are on there. So those two green lights are 10 centimetres apart.
How will that be for you? And I can get off this ledge right now. It's pretty flat.
Okay, so Dougal's suggesting that this little jelly, this Medusa that we're looking at, is supposedly a bipolar species. So by that I'm assuming that means it occurs far north in the northern hemisphere and far south in the southern hemisphere, and has never been recorded anywhere in between. Or there's no specimens from in between.
You're on one right now.
Yep, band section is on right now.
Section's on. One is empty.
It's a little dusty. Not bad. Alright, coming in.
Get 'em.
Tychogastria polaris. So that is interesting. And there we go. Collected.
There you go.
And he's in. Very nice. Indexing.
Excellent. Oh, well, it's going to be a busy afternoon in the lab this afternoon.
I don't go on. You're not here.
Yeah.
Keep going up.
Yeah. Let's keep going up.
It's like no fissure. Okay, going on right now.
Awesome. Dougal says that's awesome. Thanks very much, guys.
Stone's gonna be doing that job. It's like a really straight wall right here. Seeing that?
Wow, look at that.
The decimal. Gotta get on this side of it.
So when we saw those rock outcrop boulders just a bit earlier, it looked like they were not in situ, that they were just boulders that had fallen down, but this is looking more like it is in situ, meaning in place. So this could potentially be the base of the reef. So yeah, we would definitely want to try for a rock if you find an optimal bit that's sticking out that you think you could get.
That looks like over here.
Yeah.
Our best bet.
Yep. Dougal, yeah, we're in 707 meters. I would have thought that that's a bit shallower than the Antarctic intermediate water. Temperature is 7.1 degrees C.
Anything in here? No. Not too big.
I'll take anything. I'll take anything.
Yeah, we're looking.
Upper reaches, perhaps.
Maybe try that up there, maybe. That big thing? Yeah. Jump. What about one of these guys? Yeah, like starting there.
Ooh, look at that one right there. Sound?
No. I'm sure you did. Just putting a ram out real quick. Sorry.
There.
Is that a little eel swimming in there?
Right there.
Good.
So Rob's just joined us back in the control room. Rob, just
Barely.
20 minutes or so ago, it looked like we started to come across some boulders which looked like they might have just been blocks that have come down. But where we're sitting right here, this looks potentially like this is in situ outcrop. Either that or it's an enormous reef block that's come down. So we were just sort of cruising around looking for a spot that might catch our eye where the rock is sitting a bit proud, and we might be able to try and
Yeah, fantastic.
sample something from here.
Well, we definitely want to get a rock sample if we can. So I take it we're at the base of that steep cliff.
Yeah, we're at 710 meters now, and yeah, if this is the start of the cliff, we're just at the base of it now.
Okay. Yeah, good. Well, it looks very, blocky, massive. And I'm not seeing any strata here. Strata being layers that you might see from a sedimentary rock like, ancient limestone. It just has that kind of amorphous, massive look about it. This eel's funny, just sitting there with its tail wagging.
Yeah. Very hard.
Break. If that doesn't work, I'm wondering if there's some cobbles or something. Just see there's a little bit of a gully on the right. It looks like there are a couple of things there that we might be able to,
Wiggle something out of.
yeah. We can always crack them in half if we get them on board, but just getting something that would help. It's very vertical, isn't it?
Yeah.
Really steep.
Yeah.
Because we saw this in the 3D mapping that we've done. We've got this, so we started at the head of the canyon at around about 800 meters, and then there's this quite steep rise from around, from about 722 meters is what I measured, That was the base of this
the
cliff,
cliff.
and it goes up to about 500 meters. So it's nearly 200-meter vertical rise here.
Yeah. Enjoy your lunch, Dougal.
Yeah.
We just caught a little jelly for Dougal.
Oh, right.
One that he sees, one that's been, specimens have been collected from more or less each end of the poles and nothing in between.
Ah, okay.
So
Fantastic.
another exciting moment.
Well, I was catching lunch. I had duck spring rolls with chili sauce.
Right, I hope there's some left over because I haven't had lunch yet.
Haven't you?
No.
Oh, I thought you had.
No. It's all right.
Oh, I'll take over.
Yeah, no problem. Well, I'm going to get a drink and a bite to eat.
Oh, I didn't realise that,
No,
Marty.
that's all right. We were swapping around amongst them.
Oh, sorry. Because they did ask me if you'd had lunch, and I said you had.
Oh, I better go and get my duck
You,
spring rolls.
you better go and get them. So letting Marty have a well-earned break. She's going to get some lunch. This is Robin Beeman from James Cook University sitting on the microphone. We only have a small science team on board this expedition. Of course, lots more people from ashore, but yeah, here on board only there's 5 of us, so we all have to take turns about, have a break, get some food. Same goes with the ROV team. We have J-Rod, Cody and Chris, 3 ROV team, assisted by James on the Wirecast. So all of us need a bit of break through the day. Yeah, so J-Rod, I mean, if you do see a kind of loosish rock, if you can't pry anything off this exposed cliff wall, then yeah, we can live with a loose rock. We can always break it, assuming that it's come from this vicinity. So Marty and I have been poring over land geology maps over the last few days because we're thinking that some of this underwater landscape probably has a geological origin that is reflected on land. So we're talking really old rocks here that perhaps even Paleozoic. We're seeing a lot of quite strange landform features in the 3D mapping that we can't explain just through reef growth or old reef growth or the erosion that happens in canyons themselves. We're seeing these sort of ledges and plateaus that are quite consistent at various depths, and also some even features like plunge pools. Imagine a waterfall on land carving out great vertical cliffs. It's very hard to explain how that could form underwater, which means that we're probably talking about a really ancient geological rock that was once exposed on land in the long distant geological past, but is now through subsidence and things like the breakup of Gondwana are now stranded here on the seafloor. So hence we've been pulling out these geology maps and trying to think about what these rocks might be made of. Hence, it's very important that we try and get a rock sample. That way we can do geochem work on it, age dating, all those sorts of usual things to help us understand what this rock is made of and how it got to be here. And of course, like any good geologist, the ideal way to do it is to pull out your geology pick and snap off a bit of outcrop rock. That's always preferred, because otherwise you're just getting a like the weathered surface, which can alter, you know, through time. So a fresh piece is always good. This is a classic outcrop, and the guys are just trying to get a really good bite and see if we can lever it off. But of course, you have to remember that the SuBastian's neutrally buoyant, so as soon as you pull on the arm, the ROV is pushed in the opposite direction. So it's not an easy thing to do underwater. Yeah, Austin's saying looks very different from the seamount. I agree with you. I think what we're looking at the other day in that Tideman knoll, that was our previous dive, it looked to us like some sort of limestone rock with some hard crust over the top of it. Now, we don't really understand the crust that we saw. It was almost like an eggshell covering the softer limestone underneath. But this is quite different rock. We're deeper down. It's not a bad bite. See if we can got some pieces there. They're definitely making an impact on it. So very different geology that we're in at the moment compared to the other day. Yeah, we do have a drill. We tried it the other day at the Tideman knoll. It's quite a wide drill. It's got sort of hard teeth. There's a limitation to just how far you could probably drill into this rock. We're just going to try and snap a piece off. That might be our best option here. All right, we're going to have to do a bit more exploration. Either looking for a narrow bit of rock that's sticking out, or, you know, worst case, we can just grab a loose rock that might be sitting on the seafloor. Although it's always ideal to get a bit of outcrop rock. All right, we'll keep moving up. This is this vertical wall that we've seen in the mapping data from around about 722 meters up to 512. So we're looking at about a 200 meter vertical cliff. Of course, it's not completely sheer. There are all these sort of gullies and cracks in the rock, and lots of marine life attached to it. So this translucent stuff that you can see attached, these are silicious sponges. So sponges made of silica, nature's glass, and these colder depths, which is now 7.2 degrees Celsius. It's pretty chilly. You get these corals. These are hexactinellid coral. Sorry, not corals. Hexactinellid sponges, and a few cup corals in the overhangs there. This large one in front of us, that's called a rock sponge, so it has this kind of large amorphous mass. It's also siliceous as well. And the black coloration you see on the rock is a ferromanganese oxide, which is common in the deeper waters. Tends to coat any rock that's been exposed to the water itself. The deeper you go in the world's oceans, the more thick this layer becomes. So last year I was on an expedition to the Tasmanian seamounts out in the Coral Sea Marine Park, further east from where we are now. And when we were bringing rocks up to the surface, in places they're about, this ferromanganese crust was about 5 centimeters thick. So the original rock, like the volcanic rock we were looking for, was like a nucleus, and this ferromanganese oxide coats it with a layer, over many years. We had to actually use a rock saw to chop through these nodules to actually get to the parent rock, the original volcanic rock in the middle. But here I can see the rock face has got this sort of dark coating, and that's the same ferromanganese oxide. It's a lot thinner here though at these depths compared to, what you'd find in like 3,000-4,000 meters of water. But as we're breaking through some of this rock, you can see it's got a very red look about it. We're just trying to find like a knob or a bit of, some prominent little point that we can grab and lever off. There we go. Okay, it's still in there. Thanks. Yep. Yeah, we can live with that. Happy to take whatever we can get at the moment. Vicky, anyway.
Maybe 2.
In box 2, thanks. Yeah. So it's got a very red look about it. Typically when I see rock that red, I'm thinking iron oxide, and to be iron oxide, it's got to be exposed to air. So a bit of a mystery. We're now at around 700 meters depth. If this was once subaerial as in exposed to air, then it would have been a long time in the geological past, because this water has never been, at this depth, the sea level's never dropped this far down. Thanks, gents. Onwards and upwards, and let's see what we can find. So a blanket of sediment over any of the any gentle gradient surfaces here, but all the vertical surfaces, they don't accumulate mud or sand, and that's where you get lots of sponges and corals attached to. So this seems to be a real common feature throughout the deep oceans, and also on these expeditions. Anywhere where the rock is too steep, lots of marine life clings to the rock. So overhangs are really good. Great little microhabitats for marine life to attach to.
Hey, Rob, you want another rock?
Oh, if you can get one, yes please. Yeah,
I think we might be able to get it.
definitely looking for rocks. Did you get some lunch, Marty?
Yeah, I did.
So Marty's joined us back at the audio desk. What did you have?
You were mid-rock collection sample when
We-
I left for lunch. Did you manage to get one? I thought
Yeah,
it looked a bit hard
yeah,
and tricky.
we did. Okay, so you see these kind of these
Yeah.
little vertical surfaces that have been levering off them.
Okay, good.
We got a piece about 2 centimeters wide. It's very red.
Okay.
When we got a fresh surface, we haven't got a really big piece, but we're going to persevere.
Yeah, good.
But the red is interesting. You know, red to me says iron oxide, and you can only create iron oxide by being exposed to
Presence
it.
of oxygen, yeah.
Yeah.
Some interesting looking sponges down there too.
Yeah, so there's quite a lot of these hexagonal sponges around. These ones, we've got some longer ones over on the side. These are rock sponges, these large amorphous ones.
Yeah.
Gee, if we could get that piece, that'd be great.
Wouldn't that be great? Yeah.
It's hard though, really hard.
Cemented.
Yeah. You got the big left-hand claw. Just trying to lever it off.
Yeah.
So Cody's on the arm, and J-Rod's driving. It's quite a delicate dance.
Texas.
It's a bit of a dance, isn't it? Oh, here we go. Is that? Oh, beautiful.
Fantastic.
Well done, guys. That's a big piece.
That is primo.
There you
Oh, look
go.
at that. Oh.
It's
Nice
perfect. Keep a
sample.
good grip. Oh yeah, look at the outside of it.
Layering in.
Yeah, or maybe some boring that's going on of some critters.
Excellent.
Yeah, you can see that the black ferromanganese oxide coating. It's only a couple of millimeters thick
Yeah.
on the outside, and then there's this sort of white layer, maybe about a centimeter under that.
Fantastic. That's great.
Yeah, good job, guys.
I mean, we could just about say that's probably the first geological sample that's ever been collected in this
Oh yeah,
area.
oh yeah,
I
without
mean,
doubt.
there's
Yeah.
sediment cores that have been taken further out, deep further out
Yeah.
on the seabed. But as far as
No-
these flanks of the Great Barrier Reef,
Yeah,
that's-
no rocks have
no
ever been
rocks.
collected here, ever. And so it's more, you know, there's quite of interest amongst the hard rock, deep time geologists to try and understand, we're looking at, the continental rocks, the ancient Australian continent, now exposed here in these deeper parts of the Great Barrier Reef.
Thanks, guys. That's really good
Perfect.
to zoom in on that for us there.
Yeah, look at that. It's interesting little these holes, and these could just be, marine boring
Yeah.
animals, polychaete worms. You know, they have the ability to chew through
That's right.
all sorts of things.
So that's going to be very interesting.
Yeah,
Looks like there's distinct zones. on
yeah.
that
Well, it
face.
could just be weathering,
Yeah,
perhaps,
exactly.
but we want to get to the parent rock that's in the middle. I'm not seeing any layering there, because under a microscope you'd be able to see quite
Yeah,
quickly whether
yeah.
it's limestone or
Yeah.
or something else.
Fantastic.
Wow. Okay.
Very interesting.
Very strange. That'll be we're very keen to see that when it comes up.
All right, great.
Okay, thank you,
See if
gents.
it's-
Let's continue up.
Make some headway. What's that in there?
Is it,
Ah, is it a bivalve sticking out on the side? Oh,
yeah, a clam of some
Yeah.
kind. A big one, if it
yeah,
is.
it's big.
Couple of them.
Yeah. Yeah.
Ah, yeah, good size. Dinner sized.
Yeah. It's nice. Anyway,
We
we'll
saw
keep going.
brachiopods previous time. That deep, the Clack
Yes.
Canyons one. We
Yes.
saw brachiopods down in the depths. That was really
We
interesting.
did a few of those.
Lots of
which was
those
great
actually.
too. They're going to go to the museum.
Yeah, brachiopods are one of these kind of rare, aren't they? I mean, they're relict.
Well,
They're
yeah.
relic fauna. They thrived in the during the
Let's
Paleozoic.
just say Paleozoic.
Paleozoic period.
Covers it all.
300 plus million years ago, they dominated the shallow seas of the Earth. All that time ago, before bivalves really existed. There was a massive extinction at the end of the Permian period around 245 million years ago, and brachiopods all but died out, except for a handful of species that clung on through that time, and they live in the depths, these deeper depths.
There's some more of that vein. Can you
Oh,
see
yeah.
that, J-Rob? That vertical vein? That we were talking about the other day. If it's possible to break off a piece of that, that would be great. Yeah, thank you.
Also, there's hexagonal sponges around them.
Yep.
But yeah, the brachiopods, I think it's fascinating to see this relic fauna living there, because you can go to Chiligo Caves behind Cairns. It's an old limestone province. It's around Devonian time, and you can see limestone covered in brachiopods. And, there's hardly any of them left. The bivalves have taken over that whole ecological niche.
Yep.
But yet the other day we saw quite a few of these brachiopods in the ledges of,
Yeah, there
of some of
was a lot of them in
that
that.
strata.
In that area. See how hard this is.
Wow, it's tough, isn't it?
Yeah. Oh, there,
There
we
we
go.
go.
Where did that end up? Hopefully on a ledge somewhere.
Still rolling. Well, that's quite a couple of 100 meters to drop. No, I've lost that one. There it is.
There
Hit
it
the
is.
bottom, still sliding. Still going up.
There it is. Stop.
Just created an avalanche. Oh, it's steep, isn't it?
It's a great illustration there of how sediments move under the flow of gravity.
That's right. It just needs a bit of a
Fluid flows.
trigger.
Yeah.
So it might be here, might be just an earthquake. We don't get this. Australia's not it's outside the ring of fire. The Australian plate is actually quite stable, but it's a very fast moving plate. It's moving at about 9 centimeters a year.
Moving northward.
Moving northward. In fact, it's one of the fastest moving plates on Earth. And yet we do get earthquakes occasionally, and it might just be an earthquake that gives a jolt, and all that sediment that's been building up higher up on the continental slope just suddenly whoosh, just releases, and this is how you can end up with turbidites. So turbidites are high density currents, mostly mud, mixed with with water. And it's like this thin, fast-moving avalanche, and they deposit at the bottom of the canyons. Now we've actually seen layers of this, haven't we,
That's
Marty?
right, yeah.
The built-up layers of turbidites. So it's a big question, what triggers them? Well, earthquakes is one example of how they can be triggered. Yeah, okay. Austin's saying looks like red chert.
That's a
Yeah, okay,
possibility.
it is possibility. So chert is like a siliceous rock.
Yep.
It's more siliceous than carbonate,
Yes,
obviously.
it's,
Yeah. And when you look at it, it's
microcrystalline.
very fine. Like you can't even see the grains, can you?
No, depending on the type of chert, under the microscope, if it's a radiolarian chert, you can see the microfossils of radiolaria in there, which again is silica. So it's just, we're talking about silica here, not a carbonate.
Yeah. So that's pretty good observation there, Austin. How would you? So what's the best way to test for chert, Marty? Would you? Is there a chemical?
Well, a hardness test. I mean, it's quartz, so we know that
So
it
it's
would
really
have a hardness
hard.
of 7. It would be very hard,
Yeah.
and it would be microcrystalline, and or possibly have microfossils of radiolaria in it.
Yeah. So you could make a thin section, like
Yeah.
grind it down, take a really
Certainly.
thin piece, glue it to a glass slide,
Yep.
and put it under a microscope, a light
A light
microscope.
or a petrographic microscope.
Okay.
Yeah.
And just look for that microcrystalline structure. So looking at our mapping that we've been doing, this is this sort of grey-blue colour. This is the mapping we've been doing for the last week. And this is, we're just seaward on the
Yeah.
of what we're calling Noddy Reef. It's actually an unnamed reef, but its closest named reef is Noddy Reef. So this is Noddy Reef, and we're diving up the side of, the head of this canyon that drains this really distinct
Yeah. Yeah.
bay.
We'll have that. Thank you.
So we started around,
You want the same spot in Bioturbation?
Yeah, it can go in the same box with the other piece. It's okay. You can tell them apart.
yeah, so we arrived on the seafloor about at 820 metres, and that's sort of the big amphitheatre. And so we're driving up there, and we're now on this really steep vertical cliff. It's around about 200 metre vertical rise from, say, 700 metres up to 500 metres depth. And then we're going to do, once we get over this steep cliff, we'll be on this ledge that stretches out from the edge of the Great Barrier Reef. We'll go up as far as is safe today, so we'll be up in the mesophotic zone hopefully a bit later in the dive. For now though, we're just doing this big vertical climb up this, it's pretty sheer wall, but you can see this dramatic landscape that's to the seaward side of the shallow coral reefs, and none of this had been known about before we got here. This is the first systematic mapping of the offshore Great Barrier Reef in the very far northern part of the Great Barrier Reef, off a place called Cape York Peninsula. So Cape York Peninsula is thought of by Australians as quite a frontier area. Four-wheel drive enthusiasts like driving to the tip. If you say you're going to the tip, everybody knows what you're talking about. You're going to the tip of Cape York. It's not easy getting there. It's all dirt roads, often blocked through floods and things like that. It's quite an undertaking to get to the tip. So that's just how it is on land. Of course, offshore it's just as hard to get here. You need an oceanic capable vessel like the Falkor to be able to get into these waters. So we're very lucky to be able to get up, have this expedition with the Falkor and the ROV SuBastian. And we're calling it the northern depths of the Great Barrier Reef because, well, for a start, no one has really done any detailed mapping, seafloor mapping up in this region, and no one has ever put a deep ROV down in these waters. So this is the absolute first time it's ever been done.
This is going to go to a biobox on a quiver, right?
Yep.
Both quivers are already used.
I think it is big. We just the bottom of 3 and 4.
Yep.
So these overhangs are always interesting, aren't they, Marty? I think it's amazing that you can be in an RV, it's as big as a car, and you can just kind of, you know, just gently duck your head under the overhang to see
overhangs are
what's
always fascinating.
living there.
So it looks like we're going to collect this coral.
Soft coral?
I
We're not sure.
don't know.
I think it's too hard to zoom in, so I'm thinking when we pick it up and zoom in.
So we're just lining up the ROV now to get a closer look at it. Looks octocoral-like to me.
It does, doesn't it? Doing a tentacle count on the polyps.
Yeah.
We've got 8.
It's an octocoral.
Octocoral.
Octocoral, yeah.
Okay.
Gonna grab it?
So is that something that you want to
Yeah,
collect?
let's grab that. It's also not the whole thing.
Yeah, so we're using other communication tools to reach out to our other principal investigators ashore, like Mary Keekins, who's the curator of invertebrate biology at the museum, Queensland Museum in Brisbane. And we've had Merrick join us doing audio from Brisbane remotely, which has been really good. And we've had Jeremy Horowitz joining us through doing remote audio as well. So we're using Slack here just to talk amongst ourselves between the ship and shore, so we can get really quick updates from them whether this sample is worth collecting or not. And we don't necessarily need the entire colony, do we,
No, I
Marty?
think you're just selecting a fragment of it.
Yeah, that's right. We have limitations within our permit as to the size of the coral colonies we take, so we're quite mindful. We only take large colonies where it's really important that we get the whole structure from the basal attachment surface through to the main stems and then the branching fringe. And other times we just need a small piece, small branch, and that's quite sufficient.
Got a shrimp hanging around there. Oh, that's a good perspective on the live feed now. You're looking at the SitCam, the situational camera.
Yeah.
Gives you a bigger field of view.
Yeah, so SitCam's wider. The resolution's not as great, and the colouring's not as sharp, but it's just, it gives you a much wider perspective. So in the control room we've got, well, how many monitors have we got? It's like
I think there's 32.
32 monitors. We've got one entire wall of the control room is just filled with really large monitors, and then we've got scattered monitors at different parts of the control room. So it's pretty, it looks quite space agey in here, doesn't it?
It looks like NASA control.
Yeah, it's very cool. It's dark.
Yeah.
How many people we got? 1, 2, 3, 4, 5, 6, 7, 8, 9. We got 9 people in the control room at the moment. But of course, the ROV pilots, they've got over, I think, 10 cameras
That's
on
right,
the
10.
SuBastian itself, looking at all different directions.
Oh, that's one?
Just have a nice photo in front of the camera, please.
And would you like us to put this in the back with the other things or in its own, like in 2 things?
Sorry? Yeah, I think you can put it in the same box like for
3, 4?
yeah.
Okay, put it in the back, please.
It's been quite a sampling morning already, hasn't
Yeah,
it? And we're not even in the
we're
mesophotic.
at we've gone, what,
We've got a big job this afternoon after the ROV comes back on
need to
board.
probably
Get
make a
moving.
bit of headway, I think.
Yeah.
I know.
Well, we're trying to get up into the mesophotic at near the end of the dive. So we.
Enough image, or you want some more imaging?
I think that's good. That's fine. Thank
3
you.
or 4?
Yeah.
Great.
Can we just like pause the pod for like half a minute until the sea log refresh? The sea log somehow is not logging.
I think it just has a lag. It'll
It has a lag.
oh, has it shut down?
Yeah.
So one of the important logging computers we've got here is the sea logger, and it's like an event logger. We're taking regular photos all the time from snapshots using the science camera, but for particular events like grabbing a sample, we need to take extra photos. So,
Might want to explain what an event logger is.
well, event loggers, if you imagine, we have within the ROV we got time and position, and that's constantly being updated. It's basically taking the position of the ship, and we can use the tether to pass that information down to the ROV, and so the ROV, we know exactly where the ROV is, and we also know the timestamp. The event logger is really a computer that allows us to take snapshots where we know the position and time and depth and temperature and everything that happens for any particular event. And like an example is taking a sample. So every photo has a timestamp. We know where that time, at that particular time, where the ROV was, and so we can relate it back to a position after the voyage. So the event log is quite an important way of keeping track of samples as we go along. So we have Joan in the event logger seat and taking screen captures as we go along.
Jump in. Huh? Go. Yeah.
Onwards and upwards. So we're on the steep vertical face of this this big cliff. It's between about 700 and 500 meters depth. We're still at 670 meters. Still, we've got a good 150 meters to rise before we get over the top of this big cliff.
Yeah, we don't know what
Okay, so what's something confused?
Yeah, we did the same thing. Was you were born in Detroit?
I was born in Michigan Copper. Well, I don't know what Michigan Copper is, but it's all in the Upper Peninsula, so I don't know anything about it.
The UP?
I know that they found copper in the UP after they were beginning the exchange for Toledo, so Michigan won that. I don't know anything about it.
Yeah, so we got quite an international crew on here. J-Rod from Texas. Chris, where are you from?
United States.
All over.
I live in Florida now.
Lives in Florida now.
Born and raised in Michigan.
There you go, Michigan boy. Now in Florida. John from Canada.
Canada.
Canada. Yeah, Brisingid Sea Star. This is really obvious bright pink. It's an asteroid, a sea star. Oh, what do we got here? Some clams? Some kind of bivalve?
Rob, I
And-
think you're the only Australian in the room right now.
Yeah. We got Joan from Hong Kong?
China.
From China? Yeah. And Valerie,
She's half Hong Kong, half Belgium. So you
half Hong Kong, half Belgium. Yeah, really international crew. But check out these
James is
bivalves.
Welsh.
Yep, James is from Wales.
Yep.
The South Wales.
And I'm from God's Own.
God's Own. New Zealand.
Yeah.
Ah, look! Did you see? There's a little shrimp or something. I think is in the arms of the sea star there. Ah, there's lots. There's a lot on the wall. Have a look at all the crustaceans, all the squat lobsters and things. So this is a big a rock sponge. Yeah. And we have worm snails. These are these really obvious tubes, calcareous tubes on the side. Plus, we probably have just normal polychaete
Yeah.
worms as well. Oh, little feather star. Sea lily. So this is a stalked crinoid, quite a small one. I like seeing that.
Quite delicate, that one, isn't it?
Yeah. We've seen some really big, tall ones
Yeah,
over the last.
and bright colours.
bright
That one's-
red and things like that. So that's one of these relict animals. This stalked crinoid,
Yeah.
related to the swimming crinoids that we saw
Yeah.
earlier in dive. It was that's a comatulid crinoid. It can swim. It's not attached to anything. And we got cup corals. There's a lot on the wall, isn't there?
Yep.
Little squat lobster sitting under the rock sponge
Okay.
with this black patina, which is this ferromanganese oxide.
Yeah.
There's a lot going on there.
Let's carry on.
Yeah. But we should stick our head under these ledges and things. There's always plenty to see.
That's a nice wide shot there, giving us a bit of perspective of
Just how
the
steep
outcrop
it
that
is.
we're climbing up.
Yeah.
We're at now 650 metres. And along the
Wow.
way. Wow.
Some sea cucumbers just clinging to the rock. There's a few
few
more
more of those
feather
crinoids.
stars over here, crinoids. And there's a bit of a fringe right on the crest there. I wonder what's... Don't know.
Who knows?
Very delicate looking. It looks like snow, doesn't it? That it's just this constant rain of, well, they call it marine snow, don't they? See some
Something poking up from
feather
the other
stars,
side.
yeah. It's,
Teasing us.
yeah.
Well, this is the cleft that we saw in the multi-beam mapping. For
Yeah.
Sure.
Still, I don't think the multi-beam mapping quite does it justice. This is way
Correct.
steeper,
This
way
is
more
near
dramatic
vertical.
than any of the 3D mapping that we've
Wow.
done.
It's more than just steep, it's
It's actually overhanging.
it's overhanging.
It's overhanging.
Over-
You
yeah.
have to keep backing up.
Yeah.
Wow.
Yeah.
Oh, look at that landscape.
That's not.
I wonder if this is that bump that we were talking about when we were drawing the profile line and we kept adjusting to make sure we caught that bump
I don't think so, Marty. I think that's still higher up.
above us.
Yeah, I think it's still around that 550 metre mark.
Okay.
So we've still got another 100 metres to go before that kind of bump that we'd mapped with the multi-beam. Oh,
This is
nice.
an interesting little
yeah,
crevice.
Yeah. So this is a Brisington sea star, quite long arms, and looks like we've got lots of clams sitting behind it.
Yeah, they're beautiful.
Yeah, real blood red kind of, you know. They remind me of, you know, see there's pictures of chemosynthetic bivalves. You know, they've got that real red
In the
colour.
hydrothermal vents.
Hydrothermal
those ones.
vents, yeah. But you can you look right inside them here.
All about.
There are no hydrothermal vents
No.
here on the Great Barrier Reef. It's very stable. There's no volcanic.
It's beautiful, amazing colours.
Yeah, so bright. Yeah. So it always strikes me in these waters, it's dark, completely pitch black down here if it weren't for our lights. And yet there is a lot of colour in the marine life.
Acestophyll clams. Thank you.
It's from what they're eating, right?
Well, red, they often colour red because red is a light, is a pigment that's really absorbed very quickly by ocean water, and so if you really want to make yourself invisible, you colour yourself red.
Because that's the wavelength that attenuates first, right?
Yeah.
So to us, the red stands out, but
Only because we're
we're putting
shining white
light
light on
on
it.
if
Yeah.
not for the light, it would.
Oh, it'd
it
be
would
completely
invisible.
black here. Yeah.
I thought that very well, with all the stuff they eat is actually coming from the surface. That's what some of the old stuff is.
It's interesting how pitted the surface of this rock is. Those tiny little pitted holes. To me, that looks like dissolution, like carbonate dissolution. Pits.
Yeah.
Cast, essentially.
How much of an overhang there is.
Wow.
Like it's nothing but overhangs here.
All overhang.
Wow.
So the ROV has to keep backing going
out.
backwards to get away from the wall. Still seeing lots of these clams.
Yeah.
Oh, this is
These
interesting.
yellow things.
What's this?
Don't know what they
Can we
are.
get a closer look at these, please, Gerald? Wow, I don't think we've
that?
seen anything like this before.
Is it a coral or is it a zoanthid?
Yeah, colony of some kind. Zoanthids?
Zoanthids, maybe?
How close? You've seen it before.
They look scleractinian-like, right? So if you have a look at the dead ones, you can see the scepter. Yeah, okay, but I've not seen anything like this before. So a hexacoral, a scleractinian coral of some
Some
kind.
in Western Australia.
You did?
Yeah.
So-
They're always dead though.
Right.
So we would see the big groups of them like this.
Yeah,
in
okay.
colonies. We got a whole bunch of samples, but we never saw them alive.
Yeah, right.
So are they a cup coral, a colony of cup corals?
Correct.
Can you reach out to the guys at shore, just see if this is worth sampling? These are really interesting. I don't think.
I'm not sure how close in we can get.
Oh, really obvious, distinct yellow,
They are, aren't they?
yellow flesh. Yeah, they
So
definitely look
that's-
scleractinian-like.
Yeah. Those grooves, those ridges are what we're talking about, the
yeah.
scepter, part of the coral skeleton.
You
Yeah.
know that coral that I had on the microscope yesterday?
Yeah.
You told me to get the biology.
Get the
out of it?
biology out of the way.
There you go. That's what
That was
was.
that. And you haven't seen live ones? You've only seen dead
I've only
ones?
seen the dead ones.
No, I reckon we should try and collect it.
Never saw a lot now.
Yeah. They were like, I mean, I'm talking massive clumps of them.
Yeah,
Yeah.
okay.
Getting lots of samples of them for
Right.
Right.
in Bremer Canyon and Nebula.
Yeah.
I never saw them alive.
Well, it's definitely colonial. You can see how they bud out from the edges, like this one down the bottom.
Yeah.
It creates this. Yeah, all right. I think we should definitely
I think we
sample
should
this.
try and sample it if you can get in there. I'm not sure how close you are to that overhang
Oh,
coral.
no, we're good. It's just, these are a little tricky.
Are
We
they?
had to use the scoop to get them in. Yeah.
So the whole thing?
No. No.
Well, I mean, it's a really
It's
fragile
whatever
thing.
whatever you get, we'll just
What we
whatever
had to do
comes.
before was take the scoop and get it underneath it,
Oh yeah.
and then start coming up, and then whatever went in, went in.
Okay.
All right.
Or just come down.
Yeah, well, I think we've got a little feather star hanging out of one of the dead polyps. That's all that is. So we're going to try and get a couple of branches of this colony. But J-Rod was saying over in Ningaloo Canyons, which was the voyage run by Nerida Wilson out of the Western Australian Museum earlier this year, before the whole COVID crisis really struck, that they saw dead colonies of this coral, but no, they didn't see any living polyps in them. So this is an opportunity to grab a small sample. And there's another one just below it there, Marty.
Yeah. Do that. And then we really have to get going.
Yeah, we have to get going. We've got a ways to go. What are we now? 630 meters. We started at about 820 meters. We're trying to get up to, I don't know, 100 meters or so by the end of the dive. Yep.
And we're gonna have to stay down. Look, we can't come up early because we have a meeting as well.
Okay.
Do you want to kill it before 3?
No, because we would make it.
Yeah, okay.
So we have to come back.
Rightio.
Don't have plans.
So we're just talking amongst ourselves about things we do on board. It's just like another day at the office for many people. So we have Zoom meetings, ship and shoreside. Marty, you and I had a ship to shore with
call.
University of California in Berkeley,
Berkeley.
big marine science course. So we've done that. Yeah, so it's always busy on board here. There's lots going on.
As well as trying to keep up to date with our normal jobs back at home and all the emails and
That's right.
Zoom calls and what have you from
But it never stops,
home.
does it?
That doesn't just stop.
Yeah. So Jeremy's joined us. Yeah. So Jeremy, we're just saying that J-Rod's seen the dead colonies over in WA, but we're not sure what we're looking at here. So it's definitely a scleractinian and it's colonial, but very large polyps. Maybe you can have a bit of a dig around shore, see if you can come up with some names.
Yep, Jason in Michigan, every diver's ship to shore for you. Fair call. And all the other regulars on the YouTube.
Yeah, it's a real privilege to be here. It's hard. It was very hard getting on board the ship. COVID's locked down Queensland. It's a Queenslander only affair, unfortunately, on board. The science team could only get on board just because we were in the state where the Falkor was. We've got lots of people watching virtually. A lot of principal investigators scattered all over the globe, really.
Yeah.
But to get on board here was quite an effort.
Yes. Try and prioritise the live stuff, please. Tricky operation.
Yeah, it is.
I mean, the scoop up beside it now gives it
Tricky.
Gives you a bit more of a sense of scale now, having the scoop right beside it. 'Cause
Yeah.
I've seen the scoop, so I know how big the scoop is. So
It's a
that's-
really big polyp,
that's
isn't it?
actually really big, aren't they?
Yeah.
Yeah. Quite delicate, aren't they?
There we go.
There we go.
Okay.
Oh,
Oh,
good
perfect.
catch.
Well done.
Catch. Well done.
Slam dunk.
You okay with it just going in bio box too? Just
Yeah, you're just going to keep it in the scoop and just put the scoop away. Just going to put it in the box.
Yeah, it's going just on this box.
Yep.
That,
Just go.
that'll be the safest.
Yep. Great.
Okay, hold on, just for your portion.
Those polyps are easy as big as your thumb,
Yeah.
aren't they?
Yeah.
Really large.
Very interesting.
Yeah, if you have a look in the bio box, the E there at the bottom, you can actually see the drill.
Yeah, it's
Drill bit we
in
used
there.
the other day.
Yep.
I think the rock was so hard we were just polishing it really, weren't we?
Definitely.
Yeah.
You need a diamond drill with a core catcher on it.
Yeah, that would be great, but looks like we're just gonna have to snap bits off when we can.
Nicely done. Okay.
Excellent.
Excellent, thank you. And yes, that is a very nice pencil urchin on the wall.
Mm.
All right.
Okay,
Onwards
onwards
and upwards.
upwards, yeah. As we've been saying for a while. And then it's like, whoa, stop. There's just so much going on here. I really like these deeper waters, but of course we've got people who are keen on mesophotic depths, the shallower waters. There's a lot of life there too. So we're at 631 metres. I think we're going to top out at this at around 517 metres.
Jelly.
Oh, look at the eyes on this fish. How
Wow.
big is that? The eyes.
That is googly.
Crazy. The whole head is just eye. It was clearly a visual predator.
Yeah.
You know, bigger eyes, I guess more light.
Not sure if Will's still tuning in.
Epigonus.
Drunk Epigonus.
Yeah, probably just stunned. I mean, it's pitch black down here normally, so
Yeah.
it's just spinning around.
Terrific. Yeah, I think so.
This is good.
Cover a bit of ground.
Yeah, but if we see a really good nook cranny, let's just have a look in there. Just gonna kind of pick the speed up a little bit here. Got a ways to go, so of course if we see anything really interesting we'll stop. Wow, look how that mud just drapes. Oh, look, with one hell of a hat.
Yeah. seen a few of these now.
Yeah,
Always with a zoanthid colony on the back.
we collected a smaller one this morning,
Yeah.
didn't we? But this is a decent size. So we've got the lasers on. They're 10 centimeters apart. So this is, you know, good 10 centimeters, maybe 15 centimeters.
It's literally dragging in the mud.
It's
How
like,
can that be energetically efficient?
oh my god, where's it going to land?
It is
Oh,
acting
maybe
like
that
a
is the parachute. Oh, that is so funny.
You couldn't make this stuff up, could you?
No. Damn, it's just hanging there.
Wow. Oh well, maybe there's a method in the madness after all.
Oh, that's crazy. The fact that it can hang there in the
hanging
water.
there.
Swimming. Oh, this is just crazy. Drunk fish, crazy crabs. Oh my god. Deep sea is so weird.
Yeah.
Big sponge here, yeah. Silicious sponges. But if you have a look, these little pop outs of red, these little lobsters, shrimp, and the like. So we've got a sponge here. It's been taken by fauna. You know, the sponges or corals get used as habitat for lots of other animals to sit up on. I was just thinking about some crabs. I know like the land crabs that you find climbing up trees and things. If you disturb them, they will, like hermit crabs. They'll roll into a ball. They'll tuck themselves in and use gravity, and they'll
And
just
roll away.
roll down the slope
Yeah.
to where it was off a cliff. You got a parachute? Why, you know, they're not going to be bothered. Lots of bivalves in there. We want to have a look under the ledges when they appear. There's always lots of interesting life attached. You can see there's hardly any sediment building up here.
These file clams again.
Is that what they are? File clams.
Filter feeders, of course.
Yeah, yeah. Oh, wow. All there, Marty?
Sorry,
You're
I
pretty
was momentarily distracted, but my first instinct is no. I think
they're more
bivalves that we're
bivalves.
seeing.
Yeah. I just wanted these
see these
smaller
other
ones.
smaller ones tucked in there.
Can you just stop for a moment, guys? If we could just zoom in, just having a look at these.
Yeah, no, I think they look more
They're more bivalve?
Yep, yep.
Okay, thanks. Yeah, we got a mix of sponges, the big bivalves. I was just wondering if these smaller ones. Oh, there's a gooseneck barnacle at the top, just
yeah. See
left.
that
So
now.
this is a crustacean, a gooseneck barnacle. It's attached to the rock. So we're at 590 meters. Should be topping out around about 517. Wow. I've never seen this many in one
No.
spot before.
Coral. Just pause here momentarily while everyone gets to have a look at this coral and decide if that's a sample that's of interest or not.
It's a soft coral. It's an octocoral, yeah.
Yep.
Brachypod?
Yeah, that could be.
We got a
brachypod
brachypod
there.
just
Yeah.
behind it.
Yeah.
This really delicate, thin,
It looks
looks
very
like a
thin, doesn't
like a
it?
clam. That looks very much like a
That
brachypod.
does. Lamp shells used to be the common name for
Yeah,
them.
yeah. And they've got this really unusual filter feeder.
Yeah,
What do they
it's
call
called
it?
a loafer four.
A loafer four.
It's called a loafer four, and yeah, which is like just a tentacle filtering kind of feeding mechanism, but it's different to, for example, what you see on the bivalves. So other things that have where their tentacles are called loafer four are the bryozoans. They're called
Oh
loafer
yes,
fours as well.
yeah, yeah.
So loafer 4-8 is their mode of feeding.
So it's like a feathery tongue, isn't it?
Yeah.
That
Yeah. yeah.
stretches out. Oh wow!
Oh, look at that.
Dark, really dark cup coral. Oh, look at these little yellow ones. Small, delicate.
Wow.
Oh, there's a lot on here, isn't there?
The black one is creepy.
Emo cup coral? It's like the handy.
Yeah, it really does look like a brachypod.
It's single solitary black cup coral.
So we're just getting ourselves orientated here.
We can have a closer look at that black emo.
Whatever
cup
it is.
coral. Living fossil, that's Jason, yeah, when we were talking about the brachiopods, they are considered an informal description as a living fossil.
It's drawing in. There's a lot of encrusting stuff here, isn't there, Marty? This is not the parent rock. This would just be whatever's grown on the outside. Jason's saying, could it be a crinoid? A feather star, some kind of feather star. Yeah. I don't know, what do you think, Marty?
Julia's talking about the file shell. Trying to flick my eyes through the chat feed to keep up with, are they talking about the cup coral or are they talking about the file clams or anemone, octocoral? We've got all sorts of suggestions here. No, not crinoid.
Wow, look at all those clams.
Yeah,
Yes,
we
please.
are.
Yep. Still got a ways to go yet. And what's the time? It's just gone 2 o'clock, 2 p.m. Queensland time. And we probably about another 60 metres or so vertical rise, and we'll be up over the edge of this this steep rise here.
Then we'll probably have quite a bit of a sediment drape on a bit of a flatter terrace, won't we? And then come back.
Yeah,
up
I expect we'll see that. We'll see a lot more sediment build up where the gradient flattens out.
Might be more brachypods there, maybe.
Ooh, what's
Anyway,
that? Oh, that's just
it's
any
a shell.
kind of,
Yeah.
just an urchin shell, I think.
Yep.
Yeah. We haven't seen a live Nautilus on
No.
this expedition
No, we
yet,
haven't.
Marty.
No.
We saw them down at Ribbon Reefs, south of us. It's about 300 kilometres south of where we are now.
And we saw lots of them out on the Queensland Plateau.
In this depth range, actually. We've seen that to around about 500 metres depth.
Yep.
Between about 500 and 800 metres during the daytime. During nighttime they migrate higher up in the water column, but this is the depth zone for them. Big question is, do they live here in this part of Cape York, the far northern Great Barrier Reef? Well, we don't know yet.
Julia's signing off. Julia Summerling, off to work. Have a good afternoon.
Yeah, thanks Julia, thanks for joining us. Julia's really familiar with where we are at the moment. Has done a lot of scuba diving, expedition scuba diving is typically what you do when you come up to Cape York. It's not a place that many people get to very often. There's certainly no real regular tourism boats come this way. So there tend to be special expeditions to come this far north. Back off.
They're all attitude, aren't they?
Yeah.
More sediment now, draping over. This might be
Still rising, but we'll
Coming
probably top
on the
out pretty
top.
shortly. Probably in the next 20 vertical metres or so. And then it'll probably be this kind of more muddy environment. Yeah, sea cucumbers taking advantage of all the mud that's draping here, soft sediment, just as a veneer on top of the rock.
yeah. There's the inset picture on the screen again there now. You
Yeah,
can see
so-
our progress up the wall.
We've still got a ways to go before we get to
Yeah.
that. Oh no, we're at the
Nearly
top.
there.
Do you see it there now?
Yeah.
Yeah, we're right at that, getting at the lip, and there's that slight bump right on the edge. But from there up, it's just a gradual rise up to the base of the Great Barrier Reef, which is around about 200 metres depth.
More of those cucumbers.
Yeah, it looks like it's starting to level out now, Marty.
It's that crust again that we've seen. Crust coating everything. What are those things? These, what are they?
Oh, what's that?
Yeah, what are they?
One of those
There's one.
strange. Yeah, you saw it close up.
Yeah. Whatever it is, it's
like
not
a-
pretty.
It's like a hand puppet, isn't it? Like a sock puppet.
Yeah. Sea cucumbers. Very well camouflaged.
There's a few of them here.
Yeah,
These big
lots of
lumps.
them when you
There's
get
quite a
your
few
eye
of them. Yeah, actually, there's at least 1, 2, 3, 4 or 5 of them scattered across here.
Oh, hello, here's a crest.
Yeah, and they're all pointing down slope. I think that's significant. You know, away from where the sediment is.
Yep.
There's another big one open here. There it is. See,
There you
close
go.
up.
Yeah.
Yeah.
Could be a
Yeah,
coral.
it's a black coral. I've got my eye in
Potentially.
now.
Yep.
Yeah.
Pause here and zoom in on that.
This is what we just got.
Is
right?
it?
That's the last one that we got.
I think.
Similar to what we already get.
Yep. I think so. Okay. All right. I think we have this one. Yep. Keep going. Yep. Lots of sponges
Yeah,
in there.
these hexactinal sponges, like
Yeah.
little cups,
Yeah.
little white translucent cups.
Not easy to sample.
No. Some big cup corals sticking out.
Follow this gully up. Well, it's certainly a change now
Mm.
off the hard rock and onto
Yeah.
the mud.
Yeah, the soft mud.
So
I always liken the coral reefs to giant carbonate factories. They're just constantly chugging out calcareous sand and algae
Yep.
draping down the seafloor. So it's just this huge factory of calcium carbonate,
Is this-
limestone, essentially limestone just
Yeah.
rolling down.
is that
It looks a bit of
chunk
wood. Yeah. Yeah, it looks a bit, yeah, it's a piece of wood.
Yep.
And you'd see animals making use of it,
Lots
either for
on there.
food or home.
Yep.
So that deep canyon dive the other day that we saw that's connected to the shelf,
Yeah.
there was quite a lot of plant vegetation material in that and at the base
At the
down
base.
nearly
What
2
was that?
kilometres deep.
We were about 1900 on that, were we?
Yeah. Yeah, we saw
Mangrove
mangroves,
wood, mangrove
coconuts,
Leaves,
seagrass even.
sargassum.
Sargassum weed can only grow in shallow water.
What's this?
Something's floating past, and we got a anemone.
Anemone?
Anemone.
Nice.
Oh. Yeah, thanks, Joe. So it wasn't a black cup coral, it was a black crinoid.
Yeah, unusual.
Heliopus aldus.
Known from the southwest Pacific at this depth range. Interesting, thank you.
Yeah, appreciate that. Certainly appreciate all the help people can give us from the shore, helping us ID some of these animals, which are some of them very obvious. We're getting used to them, and others are just complete weirdos. And well, they're scratching my undergraduate marine science memory to go, what the heck is that?
Jelly, Solmicis.
You're getting your jelly eye in there,
Yep.
Marty?
Yeah, I have
You
now.
knowing your
Got my
different
eye
species?
in for stomach pouches and gonads.
Yeah. Yeah, right. I think you're showing off now.
Oh yeah, Dougal's still online. Solmicis. Knew it. Told you.
Go, Marty, yeah.
Little cave in there. These are always fun. I wonder if we'll find a shrimp party in there today.
Yeah, like we saw, there's a fish on the left.
Oh yeah. There's a shrimp.
Yeah, definitely a
Not a
shrimp.
shrimp party.
What's
Oh,
it?
there's two
It's
there.
worm snails at the back. More
Okay.
shrimp up the top.
COVID shrimp.
Yeah, there's small ones. There's a couple of little ones here.
Yeah. It's a COVID shrimp party when you can only have 5 friends over. Oh, there's a few there.
Yeah, brachypod on the right, I think. There's a few down the bottom. There's some more down the bottom. When you get eye in, there's
There
actually
you
quite a
go.
few in
That's
there.
our little stars and stripes shrimp. It looks like the flag.
There's a worm snails here on the...
Oh, there's more of those black.
Ah, yeah, these
Crinoid
black
things.
crinoids, yeah, feather stars. And now that looks like a classic
That's brachiopods, definitely. It's that lamp shell shape.
Lots going on in here. This is a nice spot to hang out.
Shall we look at what that fish was that was out on the
Yeah,
left?
there was something. There it is. Yeah.
Is it a scorpion fish?
Yeah, it looks a bit scorpion fish. It's pretty deep. What are
Oh,
we?
look at those
491
fins. They're
meters.
Beautiful. Wow.
Oh, nice shot, guys. Okay. Hopefully our fish experts can ID this. Looks like a scorpion fish. Scorpioniform, yeah.
Dougal had shrimps for lunch.
Yeah.
That's what. Did you just spot something, Jaron? Yourself?
Very pretty.
Beautiful.
When I scuba dive, this is the kind of thing that I like doing. You're just finding
Getting
a,
up
like
close.
a little cave or a nook and cranny and just slowly drop down the seafloor. Try not to stir up too much mud and just see what I can find hanging off the walls.
Yeah. What
Ooh, what's this?
have you got there? It's a candidate that desperately wants to get into a bio box. That's what it is. So this is a whip of some sort. We don't know if it's a black coral or a soft coral.
Yeah, well, I'm looking for nodes. It looks almost black coral-like to me.
So it's
Yeah, this looks
tiny,
like black coral.
tiny.
You know, you got these paired,
Yeah,
these paired feeding tentacles.
black coral. Joe's saying black coral. Well, I think we just pause momentarily and make a decision.
Do you want a snip of a piece of it?
Okay, we're going to
Just snip a
take
piece off,
snip
eh?
a piece off of it.
Yeah.
Just adjusting our position here. Oh, it's long, isn't it? Really long. Oh, the shrimp are coming out. Just maneuver ourselves into an optimal position here to snip a piece of that black coral. Wow, that is long.
What do you want?
Like a foot.
A foot, if it's stretched out, or a foot coiled.
Foot. Oh, now you're getting technical. Foot stretched out.
Alright.
sure there's a sign up around here somewhere saying that Falkor is a metric ship. So we'll have 30 centimetres of that, please.
You're talking to an American, I'm fucking way better than that.
Hold on, I gotta do the math, man. Yeah.
Alright, this is going to be interesting.
Only a foot?
Take what we get.
Alright. Yeah, put it down. You can grab it with the other arm.
Just go.
Just go for whatever you can get.
All of it.
Sorry.
Nice. Good job.
Probably put that in the suction sample.
Started in
Are
there.
y'all good with that?
Yeah.
There's a section.
Yeah, because it'd be a pain in the butt.
If wherever you want to put it
It's gonna be painted.
to tuck in,
Yeah.
you want to suck it. Okay.
I think we can put it in.
They're going to try and use a suction sample. One of the jars is going to be used different.
One please for jelly. For jelly, then we use number one.
Okay. So jar 2.
Get your dirt.
Could hand it off.
You almost got the tail end. Just bring it in.
It's almost there. Are you putting it in now? Open it up.
So what we're trying to do here is put that long piece of whipped coral into the suction sampler rather than into one of the bio boxes.
No. There, it broke though, so I got some
There you go.
So, I can put it
So
in
now
somewhere
it
else.
can go in a box.
Actually, it's in there. It's in there.
Is it? Oh yeah, it is.
It's not going quite yet.
So you just went in.
Looks like a diatom.
Oh, it's right there. There it goes.
There it goes.
No, it's inside of this. It's on.
Oh, I see.
There it goes. There it goes. Now it's in the jar. We want this little bit in my jar.
Do you want to chuck
We
it
got the
in?
long piece in too, so you need the little piece.
I'm trying to stick it on.
No, if we got it in the jar, that's okay. Yeah, but
Just turn something up for a second. Come back on with it.
yeah, it's in there. Yeah. 2.
So put it on the outside now.
Hmm?
Yeah, I couldn't see it somehow just now. Now I can
Yeah.
see it.
Yeah.
Can you grab it?
I'm trying to fly in there.
You got a lot, most of it.
Alright, okay.
You got
Great.
more than we're supposed to get.
Put it down to rounding in our metric to imperial conversion.
That's 30 centimeters, right?
Yeah, about that. All right, let's go.
Those are 2.
It's like going on a road trip or something.
Those are 2.
Anyone wants to know why we're not there yet?
Are we there yet? Is it much longer? All right. Is our scorpion fish still sitting there?
It takes me 7.5 hours to my house with these kids. It takes me 10 minutes.
All right, we'll carry on our climb up.
Look, you want up in here?
You don't.
What happened with the game?
We won.
We won?
Yep. 3-1.
You don't have to say it like that, Chris.
No, I mean,
43.
Series.
Who won what game?
Astros came back from playoffs. Baseball.
Sounds important.
It's
Baseball.
Cody, we're doing us sitting right here when I had the World Series.
where?
My mom's giving us such a
Fine.
Onwards and upwards.
She's gonna keep telling you to go up, but then she's gonna say stop.
And then she's gonna get mad at me and just stop saying, can we keep going now? 100 meters?
Right. I'll relieve you after. It's the top.
He's the top of the back.
Nice anemone.
Yeah. So nice. See you there. Here comes some rocks.
What's that? A rock. Bright white. Rock turn. A rock.
Lots of brittle stars on this one. Crinoids and brittle stars. And some sponges too by the looks of it. We'll have a quick scan around and then we'll go on our way.
Black tigers. Tiger bird.
There we go, a little bit of a ledge here. So, looks like we've certainly come to the top of our first cliff. And we'll keep going along our transect. And we should come to along this terrace and then to the base of another steep slope, which will hopefully take us up into the mesophotic reef zone. That's our goal for today, if we have enough time and if the conditions allow. Our fish. What have we got here? Oh, he's got barbels.
How's that?
Yeah, he's busy.
Oh, is that?
What fish is that? Waiting for an ID from our friends online on the YouTube.
No, he's just got like a Fu Manchu, I think.
Yeah.
Polymyxia. Polymyxia. Thank you very much, fellas.
Keep walking.
He's trying to get away from you.
And this looks like there's another one up there. Nice, another nice big anemone. And what is this little yellow guy? Busy fish, yeah.
Dead fish.
Is that another coffin fish? Nice colours.
Frogfish.
Another. Look at those green eyes, wow.
Give things away surrounding myself.
Alright, then.
Chano, I don't know how I'm pronouncing that. A different coffin fish.
Intention of eating it.
Very nice. Let's keep going.
What's the target then? 50. 50.
Yeah. Sound like you don't believe me.
We'll make it. I'll make it.
See that says keep going. That's at 280.
That's a 2-8. Okay.
Hi, Jimbo.
Hi, Big Cat. Paul. Duncan. Is he on?
Seriously.
Really? Seriously? Is it data or data? To the left?
Right there.
It is. Sure.
It's a live one.
Yeah.
Oh, there you go.
Oh, excellent. We're going to
Got
stop
one.
here.
Is
There's
it
a living one.
Is it sleeping?
Yeah, it's sleeping. Daytime they sleep, nighttime they hunt up in the shallows. Well done, guys. This is our first live Nautilus.
Uh-oh, smoke out.
And he's gone.
Looks like a magic trick.
Yeah, we stopped for Nautilus, so we'll just linger here for a little bit. So we saw two empty shells in the bottom of the first canyon we went to, off the Clack Canyons area. This is our first live one. Hopefully we haven't smoked him out.
Welcome up.
Just having a look in the other cameras. This species is probably Pompilius nodosa. And Greg Borad, who's an American researcher, he came out to the northern Great Barrier Reef and worked with Andy Dunstan, an Australian researcher who did his PhD on Nautilus, and they're still thinking there might be a couple of, maybe possibly new species or some geographically isolated group of Nautilus here, but it's a Pompilius genus anyway. We're just having a look in all the other cameras. Sebastian's got about 10 cameras on it, so we're just having a bit of a look around. Still having a bit of a search around here. So we're just trying to hold the ROV steady while the guys look in all the different cameras.
It's quite the snowstorm. Oh well, we've seen one, maybe we'll see more. Oh, spotted. Here's another coral, whip coral. There's a lobster on there. I think this looks more like a soft coral. Do you think it's a soft coral?
Wait until it's focused on the polyps.
Yeah. Need to try and do a polyp count here. Let's see what we're looking at. One, two, three, four, five, six, seven, eight. 8 polyps. This is an octocoral, so it's a soft coral. So I think we'll move on.
Move on.
Yep.
Just their nose been flying by.
So we just got the Nautilus meter going. We're up to 1. But it was pretty brief, wasn't it? It just took off very quickly.
Very nice urchin. It's got long spines. Anyway, let's cover some ground, make up some time.
We have an urchin meter.
It's about, how many is that now on the urchinometer?
Quite a few, yeah.
I like these ones, I like they've got really long spines.
Yeah, yeah. So we had a groper-o-meter the other day. That was maxing out, wasn't it?
It was.
We had those big cod. I think it was well over 10, wasn't it?
Yeah, I think we hit 11, and then I think we stopped counting after that.
Yeah.
I don't know if we were seeing the same ones again, or
But they were big
certainly at
fish,
least
weren't
11,
they?
and they were
Yeah.
big. There was one that was at least a metre. Another one of those urchins with the long spines.
We have seen them as aggregations in other places.
Anyway, let's keep going.
Don't worry, we'll keep an eye out for Nautilus. What happened was we were moving upslope pretty quick, and when we hit the brakes, it just sent too much sediment into the water, and the Nautilus snuck away. So we'll keep an eye out for sure. And we always stop for Nautilus and Dumbo octopus.
Ninjalus. It was a ninjalus. It's the one that got away. I hope we didn't disturb his sleep too much.
Yeah, it looked pretty quiet when we came across them, because they stay down in these depths during the daytime, and as the sun drops for nighttime, they migrate up into the water column around about that 200, 300 metre mark. But it's good to know they're here,
Yeah,
right? So now it's our first live one. We know that they're also on the far northern Great Barrier Reef off Cape York. Oh, one of
Another
these
one of those crazy
small-
crabs with a
crazy crab.
fancy fashion in the fields races
Yeah. So
fascinator hat on.
yep. So there's a crab under that.
Yeah.
It's not some coral that's going for a walk over the seafloor.
Oh, it's one of those shells. Is it? Oh no, is it the crab? Anyway,
Probably a crab.
yeah. Keep going.
Nope. Ray's saying carrier shell.
Carrier
Those
shell.
weird
yeah, that's
carrier
the one.
shells.
Yeah.
Yeah.
They are
They,
weird.
they are strange, aren't they? They glue other
You right down there, Ratty?
gastropods to their own shell. But these large burrows, I reckon these are giant
Could
isopod
be
burrows.
isopod burrows, yeah.
They have that classic kind of dome shape, and lots of other animals are using them as homes as well.
Polythurian. Sea cucumber.
Yeah, big pink one.
Yep. What have we got here?
Ooh, looks like Anthomastus to me.
What is this? What is Anthomastus?
It's a soft
soft
coral.
coral?
It's an octocoral.
Oh, yeah.
Maybe not. No, this is an octocoral, I
Yeah,
believe, but
yep.
I'm not sure if it's Anthomastus. It's got a really obvious stem there, isn't it?
Yep. All right. Keep
No,
going.
they're saying sea pen. Yep, possibly sea pen. Anthomastus has this more. They tend to be the polyps tend to be fatter,
Uh-huh.
and the base that they grow out on is a bit broader too. Is it carrier shell? One of those
Second one
carrier
there.
shells. I quite like these soft sediment environments. There are all kinds of things making use of the sediment.
In fauna.
No, cannot make out what that is.
It's not just a piece of reef debris, but it looks like it's got something under
Yeah,
it. There's
it's a
little
bit of
lobster under there.
yeah, it's a little squat lobster.
Yeah.
But it looks like a bit of shallow water coral.
It's
You know,
just-
some big low bait shallow water coral to
That
me.
is what it looks like. Yep.
So we're getting closer to the Great Barrier Reef
Mm-hmm.
shelf itself. You can see it quite clearly from the back deck of the Falkor. It's less than a kilometre away. So the shallow reef is really quite close. So we're going to start seeing more and more coral debris, like shallow coral debris, roll down slope here. Yeah, lots of urchins here.
Yeah, right. Are these pencil urchins?
Yeah, that's what I think of them. Pencil urchins, big, thick spines. we're in this interesting zone now, 400 meters, where the temperature will start warming up pretty quickly. So it's about 12 degrees Celsius here now. But once we pass through this 400 meter mark, which separates sort of Antarctic intermediate water.
No,
Subantarctic mode water.
With the subtropical mode water.
Yeah, so it's definitely a sort of more surface warmed waters.
So that's rising quite rapidly already, and it's 12.5.
Yeah, so it's going to start warming pretty quickly, and it's a good barrier into the mesophotic zone where we're headed. So the marine life definitely changes from 400 meters and above.
Beat it up. Hit me out. Release.
Yeah.
Move next, yeah. Keep moving.
Now the inset's showing the scanning sonar that's in front of the ROV, so even if we have complete whiteout from sediment, we can still see using the scanning sonar what's in front of us.
Sediment looks like it's coarsening, doesn't it?
Yeah, it does. There's a lot more white flecks and things. And we're seeing more rubble,
Yeah.
sort of rubble appearing. So this is where the bits of coral have rolled down slope. Yeah, I think prime Nautilus zone during daytime is from 400 meters down to 800 meters. So we've seen them within that range. We haven't seen any really shallower than 300 meters during daytime, but at night time they vertically migrate. So we're leaving the Nautilus zone behind, really.
We better check it out before we zoom on past. It looks like another soft coral. Have a quick look, another soft coral. I think. Any interest in this one, Joan? We keep going.
I think we can keep going.
Yep, keep going.
Off we go. You just got to keep climbing until we get, I think, around about 200 meters, Marty. We'll see 200 meters is really the base of the Great Barrier Reef as we know it. This,
Yeah, that's right.
The stacked layers of limestone. So there seems to be this consistent 200 meter depth that we're seeing all the way up along this northern Great Barrier Reef. And then it'll be vertical and it'll be lots of caves and things. But already I'm seeing the seafloor is becoming a lot more coarse. So larger pieces of rock, rubble mixing in with the sand.
Yeah. So when we get to the base of the next cliff face, we might just grab a push core at the base of it.
Yeah, okay. Yeah, good move.
And then up we go into the mesophotic zone,
Yeah.
the twilight zone.
Twilight zone and lots of soft corals and things like that. Not sure how high up we'll get. We've seen hard corals like zooxanthellate hard corals down at around 100 meters. 100 meters is sort of these deep water specialist. Leptocerus is a genus. Oh, look how coarse the sediment is coming now.
Yep. Let's keep moving.
Really coarse here. So this would be a mix of pieces of coral and calcium carbonate limestone. Algae.
Algae. Yep.
Like halimeda.
Yep.
So we want to keep an eye out for halimeda. The calcareous algae makes up a big portion of the reef environment, shallow reef environment where we are here off Cape York Peninsula.
Just getting coarser and coarser
Yeah,
by the minute, isn't it?
Yeah, really.
So the reef front, reef slope,
Yeah.
all that debris gets dumped.
You think it's
bottom
coming
as it
down in
washes off.
waves here, Marty? So you're getting like little
Looks like
like
it's-
shoots
yeah.
of coarse
It does look
materials.
like that, doesn't it?
Imagine like little slides, or I don't know, perhaps even like you know, if you look at the front of coral reefs, they often have these ridge and spur grooves,
Spur and grooves,
and you
yep.
get sediment concentrated within these
Between
grooves.
them.
But eventually, that all goes down particular chutes into deeper water.
Yeah, but almost 200 meters away from the vessel.
So it's just a steady climb up this incline. So we're at 345 meters. It's already 13 degrees Celsius, so the temperature's changing very quickly here.
I mean, I'm sure there's probably a lot that we could zoom in on and spend time here, but we've already spent a lot of time sampling deeper down,
Yeah.
and we've still got a lot. Yeah.
Any
there's one
halometer
metre in there
here?
for sure. Yeah.
Yeah, halometer's this sort of flake.
Just-
All these flakes, they only live in shallow water, so this is a big pile of halometer that's a bit of coral rock. But if you have a look at these white flakes in and around it, a lot of this is halometer.
Yeah. So we won't. We could linger here, but we won't. We'll keep going.
I have to wait for the ship.
Oh, waiting for the ship.
Yeah. Yeah.
Of course.
We do need the ship.
We need that.
That's where we are.
Yeah.
Started. See the models or something?
It's like an urchin shell.
Yeah. Yeah, like a heart urchin
Yeah.
of some kind.
Yeah.
When I go scuba diving, I like digging my hands in the sand of a lagoon and just seeing what I can identify in the
Yeah.
in the sediment. It's always a bit challenging, but this is pretty easy. I mean, a lot of this is halometer and bits of coralline algae.
Yeah, some shark's teeth in amongst all of that, most likely, if you looked long enough and hard enough.
Yeah, you
Find them,
would.
wouldn't you?
Because sharks being cartilaginous, this is just about the only thing that they preserve is their teeth. And we are in shark country.
We surely are.
In fact, one of our principal investigators, Richard Fitzpatrick, has made quite a career out of tagging tiger sharks not far from where we are. And we're heading that way, up
Yep.
near Rain Island. So the tiger sharks are attracted to the green turtles that nest around Rain Island. And then they... so he's been satellite tagging these tiger sharks. And if you go to the Citizens of the Great Barrier Reef website, you can find the links to the tagged tiger
interesting.
sharks, and you can see where they've visited today,
Yeah, right.
which beach they've visited today,
Go on
and
to
avoid
I'd
that
rather
one.
not know.
Yeah, avoid that one.
Rather not know.
Yeah, but no, they've got quite a collection now. I think well over 10 tiger sharks.
Yeah.
And
Do you see them following the whale migration, or is there not enough data yet to?
no,
to
not
see
the
that?
whale migration. They're following the turtle
turtles.
migration. So they do roam far and wide. The tiger sharks are travelling in the Torres Strait up to Papua New Guinea, to southern parts of the Great Barrier Reef. But in summer they head back to Rain Island. This great feeding frenzy of green turtles that come to nest in summertime.
I'm not even playing that mark bar.
Should we check that out? Sea pen, I think. Sea pen.
Yeah, it looks
Yeah.
like a sea pin.
It's obviously very audible, me snacking on Smarties.
Yeah.
Yep, Smarties.
Yeah. So, it's a long day in the control room. We have to kind of keep the energy levels up. Yeah, that's your classic sea pen look,
Yeah.
isn't it? Like your textbook drawing of a sea pen.
Very nice.
But we've seen some variation on it, haven't we?
Yeah.
Strange one the other day
that
that
retracted.
disappeared into the seafloor as soon as we went
Never seen
to
that
grab
before.
it. Great disappearing trick.
All right. Very nice.
Yeah, thanks.
Up we go.
We're at 322 metres. At about 200 metres, we should see the very base of the Great Barrier Reef. The sort of the limestone structure that we know of as the Great Barrier Reef. It'll go up quite steeply from that point. So we expect this gradient to stay about the same, but it'll get more and more coarse sediment, more rock material, debris. Think of it like a big scree slope at the base of a big steep cliff.
There's our inset again, our mapping picture.
Yeah, so all this browny blue colour, this is what we've been mapping with the Falkor over the last week, and we're the white is us, this white line, and the target depth of the beginning of the dive was around 820 metres. We've climbed up that very steep wall, which is we've seen it's a exposed rock cliff, and now we're going up this gentle gradient. But you can see the base of the Great Barrier Reef ahead of us. This is where it goes vertical again. That's around about 200 metres depth. From there on, it just continues up to the surface until we're basically in shallow coral reef. We can see that quite clearly from the back deck of the Falkor where we are now. So we're just going to continue climbing up to the base of this steep slope, and then we'll see how far we can go up that vertical face as far as it's safe.
Past 300.
Excellent. Keep going.
You down now.
Ooh, a little
Is that
skate,
here? Little flounder?
No, it's
Escaped.
a ray of some kind. I never know if it's a guitar fish or a. It definitely looks ray-like to me.
Stevie Ray.
Yeah. Yeah, this.
I don't think it's a ray though.
It's like a shovel-nosed ray or a Rhino
Yeah.
shark. I never
I
quite
don't know. I don't know my skates and rays
No,
either.
no. Yeah, it's great eyes.
Yeah, I don't know. Skate. If we sit here long enough, someone will catch up and tell us.
Yeah. And we have seen, you know, deep water sharks and rays in previous expeditions. So it's good to see one here. 295 meters. Not too worried about leaving. No, there's no electric rays in Great Barrier Reef.
Oh, there's a big
Ooh, a bit rough here.
Right.
Okay, this is like rubble, this kind of stuff, like a big scree slope. And that white, it looks
Coral.
very coral-like, doesn't it?
Wow, look at this. So this is coral reef debris.
Yeah, it's just a
Absolutely
slid
for
down
sure.
slope. Big debris field here. And this is an upside down fungia. This is a mushroom coral. So it's upside down. It
Oh, yeah.
normally lives the other way, right? So it's dome up. But that's fairly fresh looking.
It's a fresh
This is a
skeleton.
classic mushroom coral.
Yep.
Quite fresh looking. So this would have, you know, who knows, it's probably maybe only in the last few months or so.
Yeah.
It's rolled down slope.
Yeah. Great.
So
Alright,
we're
let's
at
keep going.
we're getting close to the
Getting
base of this, of the Great Barrier Reef. It's not surprising we're seeing rubble.
No, this is exactly what we'd expect to see, isn't it, at the base of the reef slope, Four Reef.
Yeah. And, you know, you can see all these white bits of rock. This is just pieces of coral, shallow water coral. It's rolled down slope. The carbonate factory I mentioned earlier.
It's like a vertical wall.
Yeah.
Right there.
Oh, yeah, right in front of us.
Up on my camera shoot.
This looks like
See it
colony,
in the
like
sonar.
a mound-like colony right in front of us. Yeah, there's all
Yeah.
kinds of things
Yeah.
here.
Oh, there's all sorts
There's
of different
another
corals in
mushroom
there.
coral.
Yeah, there's a couple of them there.
Yeah, really obvious.
Mm-hmm.
The rest are a bit hard to tell. They look a bit
Yeah.
eroded and worn.
Yep.
Getting quite steep now.
It's extremely steep.
There's another big slab coming up.
Yeah, big boulders. We're seeing a lot more boulders and things now.
It's a big chunk. That is not so fresh.
Can we zoom in on the crunchy one?
Quick look at that guy.
Yeah. Does it look like a sponge to you guys?
No, it doesn't look like a sponge.
Yeah, and we're seeing these little delicate white corals. These are stylastrid hydrocorals. We're right in the zone for them now.
Let's just zoom in on that. That one. Yeah. That's a black coral. It's got that worm tube thing that Jeremy talks
Oh
about.
yeah, yeah.
I'll just pause on this for a moment.
Oh yeah,
Jeremy,
definitely
if you're
black coral.
watching,
You want to collect that? Yep. It's in a bio box.
yep, thank you. few people signing off on the YouTube and saying goodnight. It's the end of the day for someone somewhere in the world. Not
yeah.
quite the end of the day for us. Thanks for joining us again.
Okay, Chris. Grab it.
Pretty challenging place to live if you're an animal. So much, pretty heavily disturbed here, isn't it?
Yeah, this is
Unusual to see,
energetic environment.
corals here. You look how much debris is all around.
Hello from Singapore. Just joining us.
Do you guys still need like that 1D? Can we put it in a relatively individual one?
Well, we're going to come up. We're going to. Bravo. In the Bravo box.
Yeah, so our buy boxes are getting quite full now, and we've still got the Mesophotic ahead of us.
Yep. It's going to be fun sorting this lot out at the end
you
of the day.
bet. There's a lot there.
Yeah. Very nice. There we go. Good job, Chris.
Whoa, whoa.
Whoa, whoa, whoa.
Don't be scared. I'm scared. A little unnerved.
Keep going.
Yep. Let's keep going.
Alright. Not stop until we hit the wall.
Got it.
Hold it.
Not
Getting
stopping before we hit
steeper.
Very much.
Yeah.
Jeremy wants to thank you guys. This is a new species he didn't collect the last time.
Here
it.
for
Oh,
you, Jeremy.
terrific. Thank you. Not
Wow.
sure if you heard that. That was another new species that we didn't collect on the previous voyage. So that's another one for the collection. That will be going to the Museum of Tropical Queensland in Townsville.
Yeah. Interesting little gully there.
Yeah.
All of this rubble would be shallow water coral.
Yeah, for sure.
It's interesting is in these streams of debris. It's all focused in particular points. See more fish?
Do you want to, yeah. If you see any more of those things that look like a bottle brush like that. Maybe that one there.
Next to that sea pen.
Zooming on that.
There's also one over here.
Looks like a soft coral.
We can move on.
Yep. Thanks. Okay. Some of these blocks don't look fresh though, they look old, don't they?
Yeah. Yeah. So, you know, the core, the longest core through the reef is from Ribbon Reef 5, which is south
Yeah.
of us here. And they got
Bottomed
down
out
to
at this
around,
depth, didn't it? 240?
yeah, it was around 200-ish meters, 240 meters, where they saw a transition from sort of temperate kind of marine life through into, you know, really clearly tropical corals around about that depth. So imagine what we're on the edge of that Great Barrier Reef, looking almost like a cross section through all that. So they're looking at ages of around about anywhere between 500 to 600,000 years ago when that transition occurred. It would take a few years. You get a little bit rubbery with your ages when you go back that far. But you know, if this has been here a while, it's quite possible that we're looking at the very origins of the Great Barrier Reef.
That's right.
When it first started turning into limestone, shallow coral reef.
It's broken.
Well, that's interesting.
Yeah. Is that some type of gorgonian?
Yeah. Yeah, very delicate looking.
Very small
Oh, that's a
colours.
really interesting one, isn't
Yeah.
it?
Very small. Gorgonian, maybe?
I think it's a black coral.
Yeah, I see your comment there, Jeremy, but we are pushed for time and space in the bio boxes, and we are aiming for the mesophotic zone. So we are going to push on. We've collected a lot already so far today.
Yeah, we're nearly full. So we just have to keep pushing up.
Yep.
In a bit more on polyps.
Oop.
Cute.
And of course we need to keep an eye out for our pygmy seahorses, don't we?
Yes.
We stop for pygmy seahorses. But that'll be a bit higher up if we get there. That'll be probably around the 100, 150 metre mark.
Yeah. Jeremy, this one that we're looking at now, although it has broken off, it looks like it's very fresh. Is this something that you'd like us to put in the bio box while we're here, and then we need to keep going? It looks like the whole colony is already broken off. Keep going? Okay.
We're at 18 degrees now. The temperature's
we are.
rapidly warming.
Yep. I reckon it won't be too much further and we'll start getting our first glimpses of our encrusting coralline red algae.
Yeah. We've seen that in other parts of the Coral Sea, really clear waters around about 220 metres. So we start seeing these little patches of red on the rock, then that's red coral and algae. So we're crossing into that zone very soon.
So I think we might have quite a bit of a lag here, Jeremy. Sorry, we've already moved on from that one.
There's a crayfish just under the rock here. You just see his feelers, they're quite a decent size too. I'm just going to reach out to Tom Bridge from the Museum of Tropical Queensland, who's our mesophotic coral expert. See if he can join us on audio.
We're getting high penetration.
Wouldn't surprise me.
Oh, there you go,
Yeah,
mesophotic
we've got
zone.
some light, natural light.
Yeah, so this is with everything turned off except the lasers. You can see there's sort of this blue twilight. So very limit of light here. For me, the giveaway will be when we start seeing this red algae appearing
Yeah.
on the rock. And then you
It's
know,
always the first sign, isn't it?
yeah, I think the lower limit is like 1 degree or 1% of the available light, and that is the really lower limit of light when you see that red algae. Yeah, out in the Coral Sea, away just a bit further out from where we are, where the waters are really, really clear, we're seeing that popping out around about 220 meters. So
Yeah.
we're definitely within that, starting to get within that photic zone. Yep, Tom's next door. Okay, you're seeing the red on
There
the
we
rock
go.
here?
Yep, there it
So
is,
this
actually.
is that. It's photosynthetic, like it's got pigments allowed to photosynthesize, so there's red encrusting algae. We're going to see a lot of it. Nearly every bit of rock will have this red algae on it, harvesting whatever light it can get.
Yep.
Straight up and down.
Great. Okay. Keep going.
Yeah, oh, more fish.
More fish now.
We're at 20 degrees Celsius, so it's warmed up really quickly.
Yeah.
It would be quite comfortable for swimming in at this depth, if you could get down
For
there.
10 minutes.
Yeah. The
I'd
surface
still
waters.
be putting a wetsuit on.
Yeah, yeah. Oh, yeah. You lived in Queensland too long then. 20 degrees. Oh, yeah. But the surface waters are around 26,
Yep,
27
26.
degrees Celsius. Yeah, it's very comfortable here at the moment. We're heading into summertime here in in Australia. Okay, starting to see lots more soft corals.
Mm-hmm. Oh, so there's fish in there.
Can we just have a look at the
That's a
fish,
nice,
guys?
Nice wide angle there of the slope that
Look how steep that is, eh?
we're climbing. Yeah.
Well, we've mapped it. It was very steep when we mapped it.
Yeah.
It just seems like this 200-meter vertical cliff.
It's nice to get that wide angle view.
Yeah.
Sorry, I had to
That's
make sure my
alright.
boat was up here.
No worries.
Fish in here, little scorpion fish. Lots of sediment draping any flat surface.
Yeah, we just passed through Thermocline. Did you see that shimmer in the water? Did you get a sense of that? There you go. We just jumped 3 degrees. We got that shimmer in the water of the Thermocline. That was it. 171 metres. Just keep climbing.
So, notice all the ledges, the layering,
Yeah.
the different layers of reef strata build up on each other.
So this is reef framework.
These little delicate white corals are stylasterid hydrocorals.
She doesn't.
This is pretty. I guess these
Cody,
are,
do you see any caves off to the right or left?
We do not. There's a big gap there. No big cave gap.
okay.
We'll go cave diving.
Yeah. Yeah, we'd like to go cave diving.
As long as we're going vertically at the same time.
Yeah. It's getting very busy now, isn't it?
Yeah.
All right, these
Gonna-
little, these small white
there's
colonies.
That water.
Yeah. Oh, that lovely, that shimmer. So there's a
Yeah.
real change in temperature here.
It's 22.
It's 22. It's really got warm very quickly.
Yeah.
It's nice big fan corals over on the side. Are you able to track to the right a little bit? We just want to have a closer look at some of these soft corals.
Wow, look at that.
Look at that shimmer. It's amazing.
Yeah, right here.
That little shimmer right there just jumped us up to 24. And then when I got out of it, I think I got 22, 23.
There you go.
Yeah.
It's a 2-degree difference.
Yeah,
just
so
there.
yeah. So we've got two different water masses that really distinct.
Yeah, right here.
Did you want to stay on those or just get them all in full view?
We have a bit of time. Yeah, if we see these really large colonies, it'd be good to have a bit of a scan over them for any, pygmy seahorse. Oh, that's pretty, isn't it?
Beautiful.
So J-Rod's usually our seahorse spotter. He's got very good eyes.
Colours are so vibrant, aren't they?
Yeah. All right. Thanks very much. A little crustacean, or could even be a snail, just feeding,
Snail.
because they can feed on corals. Okay, we're getting sea whips coming in, a lot more fan corals.
So we're certainly in the mesophotic zone now for sure. It's just going to start getting busier and busier.
Mm-hmm. Yeah. There are all these little micro habitats left by the different layers are just perfect hidey holes for sponges. Here we go. These are interesting. To the left, this yellow colony here.
don't know my corals well enough to know what that one is.
Yeah, I mean, you do see these when you're scuba diving. These,
It's not Tubastria.
I think it is
It's
Tubastria. You know, jewel corals. They're quite beautiful yellow corals that you find them in caves. That's what they look like to me.
Alright, keep going. I think we've got a good chance of making that sort of 80 metre-ish
Mm.
room today. Beautiful colour. Let's keep going.
Oh, some big fan corals here. Big gorgonias. Well, this is the pygmy seahorse zone. It's where we've seen them before, this depth.
Beautiful crinoid waving at us. Is he going to jump off and go for a swim?
Snail shell, snail down
Yeah.
here.
Nice little community there.
Such a lot of stuff living on it.
Yeah.
There's gotta be a seaward on there.
Lots of different things colonising this Gorgonian fan.
Can I just say hello to someone?
Yeah, of course.
So I just heard we are streaming live apparently in one of the CT scanning rooms in Brisbane. So hello to all the patients sitting there waiting for their scans, and I hope this is something that's nice and relaxing in the waiting room. And hello, Kerry.
Is that your sister?
Yeah.
Hi, Kerry.
And hi, Dad, if you're watching too. think you were at lunch, Marty, when we booped one of the swimming stars and watched it swim for a while. Oh wow, that's huge.
Nose.
Wow, look at the size of these two.
They're enormous.
Wow.
Wow. That's huge. And certainly different to what it was 15 metres, 20 metres below.
Control good. Hey, big guy.
It's quite interesting seeing that thermocline. We learn all about it and know that it's there, but to actually see it with the ROV and how different it is in just such a short period of time and that actual visual that you can see with it, it's quite amazing to be able to see that change.
Are they nudie breaks?
Oh yeah.
Not sure. Not sure if it's a nudie break.
Don't know.
Certainly a lot of real estate being taken up there, isn't there?
Yeah, there is.
Maximising every space.
Yeah.
I'm gonna knock that thing over.
I'd say it's one of the biggest fan crawls we've seen. I think.
That's so big.
Wow. Here we go. It's just more and more now.
Yeah.
I'm trying to find a place where there's not too much coral.
Yeah.
Mm.
Don't destroy it, don't enjoy it.
Dang it. You know what's weird?
It's interesting that they're in a line.
In a
Yeah.
line.
They're in a line.
All in a line.
Yeah.
All on the line together.
Yeah.
All down.
That is interesting.
Mm. Yeah, that's fascinating.
Oh, black coral. Is that black coral? Yeah.
It's a very big one. It is. Wow.
Looks like black coral.
Looks like the one that we brought up on a last expedition. You had to carry it on the outside of the.
Oh, right.
We hung it upside down in the wet lab for a while.
Yeah.
now we're starting to see more fish.
Mm-hmm.
We're gonna look right up in here for.
Stabilising ourselves here, and we're going to break off a piece of this rock here. Check it out and see what it is. The texture of it is interesting to us. It looks like it could possibly have a microbial crust coating on it. I don't think a lot of that red stuff is rock.
No.
Yeah, that's good. Can you reach that?
That was a little easier than the last rock we sampled.
Great. Yeah.
Where you gonna put this one?
It can go in. Just easily chuck it into.
Yep.
Yep.
Shoulder it up. And. Shoulder up, hold that. Go to the pitch.
So this depth is pretty significant, isn't it, Marty? It's where it's about where sea levels were during the last ice age.
Yeah, it's probably a bit below. But it was just that bobbly texture on the rock caught our eye. We'll just grab a bit of that while we're here.
Yeah, certainly one of our biggest sampling dives we've had on this expedition so far, isn't it?
Yeah, for sure. Yep, something for everyone. Yeah, hoping if we get up around the 80 metres or so and it levels out, then there'll be a whole other. The habitat will change again completely. But,
Well, we don't know. It might be
at
just
all.
sheer
It might
vertical,
just- yeah.
and it may not level out at all. Yeah.
Treasure chest.
Whether we get that far.
Moving on up.
Yep.
Trying to get to the top, right?
Yeah. There aren't a great deal of fish around, are there?
No, that's right. Yeah. No, we've seen some smaller fish.
Maybe they're higher up.
Which is a ton. Should you get guys ready for this expedition?
A lot of caves around here, Marty.
Yeah.
This zone, and we've seen it on other parts of the reef where this sort of 130, 120 metre mark. Lots of overhangs and kind of more cave-like. Yes, so some of the patterns we've seen on other sheer sides of the reef is that around this 120 metre mark, 130, 120 metres, we're seeing lots of caves forming. So we think that this is indicative of sea levels during the last ice age, which dropped down to around about this level globally. Right, so not just here, but right across the globe, down around these steps. This is a black coral in front of us. Pretty sure of that. Yeah, and of course those caves create lots of microhabitats for corals and stellastrid corals and the like. But yeah, this seems to be a pattern that we're finding out in the coral reefs on the Queensland Plateau, which was our first expedition out here, and also on the this edge of the Great Barrier Reef as well.
Really want to see a horse, bro.
Yeah, I do. Thanks, Cody.
Really want to see a horse.
Yeah, all right. Well, let's linger here and have a bit of a browse while we're waiting for Tom Bridge to join us. Yeah, so let's have a good look in amongst the branches of these big Gorgonian fan corals. There's a lot of real estate here, and you just look for something. The seahorses are tiny, maybe only 5 millimeters, centimeter across, and they will likely be the same color as the corals itself, so orange or red. And we spotted the last one through its tail. Its tail was wrapped around one of these branches. Yeah, lots of shrimp in amongst the branches here. Little feather stars. Quite a few little feather stars. Just looking for anything that might resemble a tail or bobbing around behind it.
Rob, we're going to try and bring Tom in the next sort of couple of minutes. So while we're organising that, we're just probably going to cut the audio briefly
Sure.
while we get Tom set up and sort that out. So if it all goes quiet for a moment, it's just because we're changing the audio setup for bringing Tom Bridge in on through Zoom.
Yep.
Which one do you want to bring? You look like you need a break.
While we're waiting, we can maybe try for another rock for a LGM, 20 metre LGM rock.
Yep.
While we're waiting to sort Tom out, we might try for another rock.
Okay.
At this 20 metre mark. And it won't be able to go in the same box as that big one.
Okay.
Open box G, so we'll see what's in.
Did I miss? Vertical wall.
Yeah, very eroded here. Lots of overhangs, caves. Became quite distinct as we went up from around 130 metres up to this depth. So quite likely we're looking at what would have been a wave-cut platform and fully exposed to the air during the last ice age, which was around 20,000 years ago. So we're just trying to bring Tom Bridge in from the Museum of Tropical Queensland, who can talk us through some of this mesophotic coral environment that we're looking at. At the same time, we're going to try and get ourselves a rock from this this pretty significant depth zone. Just going to position Sebastian here so we can get a grip of some rock. There we go. It's a nice bit of limestone rock that we can get from this last glacial maximum or lower sea level rise.
Oh, it's straight down. There you go.
You guys have a preferred headset?
Yep. Okay, no worries. We'll wait for Tom to come on. Yep. I'm happy here. Bring him on whenever you can. He's in the waiting room now. So we're going to have Dr. Tom Bridge from the Queensland Museum of Tropical Queensland join us for audio this last part of the dive as we head up into the mesophotic zone. So we'll just store this bit of rock from this pretty significant depth, around 120 metres, which is where sea levels dropped during the last ice age. So it's always good to get a sample at these really significant depths, and we can do a whole lot of geochem work and age dating on them. Okay, we're good to go here. Tom's ready.
Hey Rob, can you hear me? How you doing?
Yeah, Tom, can you hear loud and clear? Do you want to introduce yourself?
Yeah, no problems. So I'm Tom Bridge. I'm the Senior Curator of Corals at the Museum of Tropical Queensland and a Senior Research Fellow at the Centre of Excellence for Coral Reef Studies at James Cook University. And I'm very distracted by an incredibly large looking Anello gorgonian just to the right of picture right now. That's beautiful.
I'm not sure if you were watching, probably about 15, 20 minutes ago, Tom, there was just this huge gorgonian. It was one of the biggest I've ever seen.
Yeah, no, I missed it. Unfortunately been in meetings most of the day for various things. One of the good news, I guess, to come out of some of this, we had a couple of the meetings were with the CEO of the Queensland Museum has come up to visit for various things from Brisbane, and some other important people, and we had to give a short presentation for the CEO about some of the research we were doing. I brought Jeremy along with me because we were specifically focused on some of the stuff we collected on the Falkor and talking about these expeditions, not just this Edge one, obviously, but the previous one that Jeremy was on, and highlighting all the amazing things that we've been found. So, yeah, look, it's been incredibly beneficial in terms of, you know, understanding what we have in the sort of deep waters off Queensland. It's, you know, we've gone at the Queensland Museum with Jeremy's black corals from having not very much in a very poorly known group to having, you know, one of the best collections in the world, probably the largest collection of black corals behind the Smithsonian. So, yeah, wow, it's been incredible. I'm just looking at this footage now. These look like mainly like Anella. Now, these guys are quite common, Gorgonian, but this is a really high density of them. I guess this habitat is fairly typical of a lot of this northern outer shelf of the Great Barrier Reef, and also I guess a lot of these sorts of very steep faces with these sort of ledges, notches cut in them, are quite common throughout a lot of the Western Pacific. We see these in the Pacific Islands and things as well, but they're really quite stunning habitats. If there's enough current flowing, these octocorals really love this sort of habitat where there's not too much sediment accumulating, partly because of the steep slope. So some really stunning biota here. It's both octocorals. I can also see some sponges. Sure, there's some black corals there as well. Wow, this is really stunning.
Yeah, I agree, Tom. I mean, we're starting to get used to seeing these big fan corals, but this is one of the better collections I've seen in one spot.
Yeah, absolutely. I mean, I guess there's sections of Osprey Reef that look a little bit like this. As I said, we have a colleague, Sonia Rowley, at the University of Hawaii that does a lot of work on these Gorgonians in mainly using closed circuit rebreathers to work on these, but quite extraordinary, the diversity of these things down there. So she does some very advanced diving, often down to depths of 150 metres or so, and has this amazing collection of these guys. So a large number of these species would be undescribed. So it's a very fruitful place to work. It'd be good if we can just have a bit of a look on the edges of a few of these ledges, if that's possible at all. Just like right on the edge, there might be seeing a few more corals and things like that.
Yeah, we just turned the lights off briefly to show people just how, I guess that classic twilight zone is just this filtered blue view to our eyes anyway.
Yeah, absolutely. There's not a lot of light down here, as you would imagine. So we're probably talking about, yeah, probably well below 1% of surface irradiance, probably below 0.1%, depending on how clear the water is. But yeah, it's a huge amount of life. Almost all of these things, apart from probably some of the algae, maybe the odd coral, is azooxanthellid or completely heterotrophic. So these guys don't rely on sunlight at all. They don't have zooxanthellae. Interesting school of fish hanging around this guy, this little fish. So yeah, quite extraordinary, the diversity of some of these guys. It's also super interesting. We saw a lot of these sorts of species in the AUP image from the Southern Surveyor trip that Rob, you were on as well. It's the main part of my PhD. We did do some collecting, but being able to see a lot of these species in situ, but in really high quality images, has been a really interesting feature of this whole expedition.
Yeah, that's right. That earlier expedition just used sort of grab samples. We had no idea what it looked like down there. We just had to imagine it. But we had 3D mapping from the multibeam. But we actually brought some of these up to the surface. It's really something else entirely to see them in their living habitat with this high quality vision.
So a lot of these small damselfish and things, I know a number of colleagues that would be getting very excited by these. I guess that's one of the really interesting things we've seen in the Coral Sea. There's a number of these species that have been discovered, often from the Central Pacific. There's been a lot of work in the Central Pacific, I think largely because a lot of the researchers that work on a lot of these fish and have the diving skills to be able to dive in these habitats to collect reef fish are based in Hawaii. Not all of them, but a lot of them. And but we're finding as we keep doing more of this work that more and more of these fish seem to occur in the Coral Sea and on the Great Barrier Reef. So probably much more widespread than we thought. So yeah, that's been really interesting. Really interesting aspect of this. I won't pretend I'm good enough at fish IDs to be able to ID them from just quickly as they dart in and out of holes. But I'm sure if you went diving down here, there'd be a lot of interesting fish that you would be able to catch.
Yeah, I don't think we've been overwhelmed with the number of fish here in this spot. Certainly in other parts of the GBL, we've been a lot more fish. Wow, look at all of these fan corals here.
Yeah. Yeah, look, it's incredibly impressive. Like the size of these as well. Beautiful big wall. I was going to say as well, Rob, we found some of the more gently sloping areas further to the south, so around Hydrographer's Passage and places like that, this sort of 90 to 110, 120 metre depth range was really a real peak of diversity, particularly for these Gorgonians. Yeah, partly with Hydrographer's Passage, because we were looking at series of fossil reefs. We thought that may largely be due to just habitat availability, but it's certainly interesting that we're seeing, you know, these aggregations than what we see here. Sorry, there still, Rob?
Yeah. We're just quietly, just taking it all in. It's someone says sideways forest. I totally agree. It's really amazingly dense.
Indeed. Yeah. Sorry, I lost you for a second. I'm zooming on my iPhone as it seems to have much better audio quality than my computer does. Yeah, the way that a lot of these Gorgonians, we see this, the way that they're, as you mentioned, the sideways forest facing straight up and down. This is something we often see, we've seen before in the northern Great Barrier Reef. What this suggests to me is that there's a lot of upwelling. So these guys tend to face perpendicular to the current, and places where you have them facing as they are here, you'll often notice that the primary currents won't necessarily be along the reefs, but they'll be straight up and down. So you can get really big sort of upwelling induced currents and tidal currents coming off the reef that cause really strong down currents at particular times. So they can be very hairy places to dive. Down currents aren't very pleasant for anyone that's been diving on one of these sort of outer walls when you encounter one. But these Gorgonians, they love them, and I wouldn't be surprised if this is sort of what's the sort of habitat that's going on here. It obviously is probably pretty calm at the moment, but also the way that they're growing combined with the abundance of them suggests that, at least at times, the currents here would probably be pretty strong. If we have a look on the top of this ledge.
Yes, we saw it too. This looks like Leptocerus to me. These sort of flat-topped, dark brown corals. What do you make of this there, Tom?
Yeah, absolutely. No, I'd agree with that. There's a few interesting ones that, if we can have a bit of a close look, there look maybe some fungids in there.
I'm not seeing any fungids. Yeah, I'm not seeing any fungids. These look like your classic
one of the ones
Leptocerus.
Okay, we'll have a look. One of the ones on the sand, possibly. Obviously, you guys probably have slightly better vision than me. There's clearly some Leptocerus in there by the looks of things. There's a number of interesting corals that tend to occur in these sorts of habitats. Fairly specialised or very specialised to these deep habitats, so not usually diverse communities, but they're usually, we've seen in these previous trips, number of different species. There we go. So yeah, that guy's clearly one of the Leptocerus species with the sort of dark blotching. Can we have a look at the one just below that as well? Yeah, okay.
Yeah, Tom, it was so obvious. We went up that really steep wall, lots of Gorgonians, and as soon as it kind of angled out and became a bit more flat, suddenly there was a bit more horizontal surface, and these all popped into view.
Yeah, absolutely. So these guys, there's another Leptocerus, obviously being corals, these guys do need light. Yeah, well, what's that? What that one is. Can we have a look just to the one to the right as well, of where we were? Oh, there's a regal angelfish, Pleiades diacanthus. They probably like these sorts of habitats. So yeah, just
there's a mushroom coral.
Right, it's kind of hidden. Yeah, there we are. Yeah, no, there is. We saw one there. There are a few of them down there. They seem to be pretty.
Yeah, we saw one of these, like a dead one, down in the scree slope at the base of the GBR, so quite fresh looking, down around 200 metres. But yeah, we've definitely hit the zooxanthellate coral zone. We're in 87 metres of water here.
Jeremy will be being upset that we've got we're looking straight through a black coral there. see this fungi.
Yeah.
But, yeah.
to the mushroom coral on the other side. For you, Tom, look, we can sample here. So it's your call how we do this.
Yeah, look, I guess I'd be interested. Can we keep going up a little? I'm really interested to see what's around here. I've got another few minutes before I have to tug off, and it's very surreal, this whole experience that watching these ROV videos of this unexplored deep Great Barrier Reef, and then I've quickly got to run off and go pick up the kids from daycare. It's definitely a very different way to do science that I never would have envisioned before this sort of COVID. Okay, there's a few little episodes. There's one here. If we just zoom in, there's one in the middle of the screen.
I was going to say there's some very wide ones here.
Yeah, no, there's quite a small one, which I think is a Leptocerus, unless it's a sponge, just to the right of that one. So that one looks like one of the encrustance type ones. If we can go to the right, it kind of looks squashed in a hole. Just have a look at that. It might. Okay, no, that's a prize. That one actually. That's interesting. There's a particular one that I've been looking for, which is a new species which you collected on a habitat like this in Micronesia, and currently in the process of describing it. I think we may have got one from the Visioning the Coral Sea, but I'd certainly be interested in trying to find more of it. Okay, so there's
some really delicate little ones as well. Yeah, with the white sort of rim around it.
Yeah, can we just zoom in on one of those? I think that's the one that we would probably these days be calling Leptocerus fragilis, most likely. May not be that species. We've really got to do some detailed taxonomic work. So we have a colleague, Francesca Benzoni, who's in Kaust in Saudi Arabia at the moment, but really world-leading coral taxonomist, who's been doing some really interesting work on these guys. So hopefully some of the ones that we'll be able to collect. Oh, yes. Yeah, see, wow, look at those beautiful striations on the septa. So I might grab that one if that's all right, if that's easy to get.
Yeah, roger that. We'll set ourselves up here.
And we're seeing getting close.
Yeah, that's right. I was just a shout out to the Halimeda lovers amongst us. The green algae. These are the fronds, the sort of cornflake stacked fronds that are hanging down. So this is coralline green algae.
It'd be great, I guess, with some of these Leptocerus if we could potentially snap freeze them in the liquid nitrogen, because they're really towards the lower depth limit of where hard corals grow. I mean, I know we did find, I think before this work on the Falkor, the deepest we'd ever found zooxanthellate corals in Australia, at least in Eastern Australia, was about 125 metres. On the previous trip, we found them down to over 140 in the Coral Sea. So it would be very interesting to start to see, I guess, trying to work on the communities of these corals living right on the edge of their sort of physiological range. There we go. Yeah, they probably won't be. Oh, it's so fragile.
May have lost that one.
Yeah.
Haven't given up. J-Rod's in control, so we might get it. There it is. It's just, quite a very busy surface here.
And these guys as well, they don't require very much, they really don't require very strong skeletons for the most part, because there's not much wave energy or anything down here. So they don't really bother investing much energy. I mean, you can imagine if you're a Leptocerus coral at this depth, you're not getting a lot of light energy. So, you know, you've got a limited amount of energy to put into, you know, making sure you do what you need to do. So whether that's growth or reproduction or whatever else. So a lot of these deeper corals, oh, look at that, skills. Well done, let's get it in. So yeah, a lot of these guys find very, it's almost like paper, these skeletons. It's they're horrible to try and transport back when we've collected them overseas and things like that. Because they're almost this sort of crunchy, papery stuff there. It's hard to believe. It's so different from a normal coral skeleton, but I guess that's because a lot of them just have no need to, they really have no need at all to be protected from waves or anything like that.
Yeah, that's right. So it's an energy issue. Like, there's just not much wave energy down here. And so they don't need to make a robust skeleton. Is that what you're saying?
Yeah, I mean, no one's ever tested it, but that would make sense from a biologist's perspective. The other thing as well, I guess, you know, where you do have such a limited energy budget, you know, I guess that's one of the things you can probably afford to let slide. You know, theoretically, if you grow really slowly down here, it probably doesn't matter because it's a fairly stable habitat. But at the same time, having said that, some of the work we've seen more recently, or done more recently, not we, but we being scientists, suggests that they actually grow faster, I guess, than we'd expect, at least in terms of linear extension. So this is work by Sam Kang and colleagues in Hawaii, and showing that, yeah, really, they grow faster than we'd expect. So yeah, quite interesting. Is it possible to go up at all, or go back down? Sorry, go ahead.
Yeah, we'll go up. Quick question to you, Tom, is do you want that last Leptocerus snap frozen liquid nitrogen, or is ethanol?
Yes, please, that'd be good.
Okay,
yep.
Roger that.
And we'll send that one over to, hopefully over to Andrea at the Smithsonian. So yeah, there are a few very interesting corals that do occur at this depth. I'd love to try and see. None of them have been confirmed yet in Australia, but all from the New Guinea and the Central Pacific. So some very interesting corals. So yeah, let's see what we can find. I'm also as well, in my particular position, I'm particularly interested in the staghorn corals, Acropora, and we did a dive earlier in the trip, trying to find some of these deep water ones that live in Halimeda fields and stuff like that. Unfortunately, we didn't find any on that trip. This is probably a different sort of habitat, so I wouldn't expect to find them here, but you never know. And again, these habitats are so poorly known that it's really quite,
more Leptocerus.
Quite a surprise to, you never know what we're going to find.
Yeah, that's
Sorry,
right.
go ahead.
Oh, just admiring the Halimeda, these curtains of Halimeda. Of course, we see the sediment all over the place, but, it's great to see the living stuff, the green curtains of Halimeda hanging down on the edge here.
Absolutely. Yeah, plenty of corals here, so Leptocerus, and probably a few other lobophiliids, so things like Carniphilia and those sorts of things around as well. They tend to be characterised by much more coarse septa. So this one we've just seen going up here, this is Carniphilia. Again, pretty common in the Mesozoic. Can we actually just go back and have a quick look at that guy? Down a little. Yeah, so there's two just in together here. All right, yeah, these two. So there's one that's sort of brown with a big central corallite and sort of a greeny centre, and then there's a little plate just next to it. See, this is one of the tricky parts about this this sort of work, is that we do have you know, a lot of the identifying these corals requires real close-up looks at the skeleton, and yeah, oh, yeah, okay, yeah. So that's amazing footage. Yeah. So this guy, yeah, this would be kind of Philia, probably Oxyphora. I bet if we tried to collect that guy, that would just crumble, and you'd end up with a big, slimy, sticky mess of nothing. But that guy just below, so just down to the bottom, is that? I don't even know if that's hard or soft. It's incredible looking. Collected something very interesting that looked a bit like this on the last trip. I still don't even know what it is. Can we grab that? Yeah, that one there. Can we grab that?
All right, see what we can do.
Wow. Yeah, that looks like the thing that we got on the last trip. It took me a little while to work out when I got it back to the museum whether it was even a coral, which is always a good start.
It's certainly delicate here, Tom. You can see it's starting to bend just as we put pressure on from underneath.
Yeah, wow, it's almost looking, if it's a sponge, we might leave it. It's almost looking like it's soft. You just poke it. So again, it's very hard without being able to see down there. We always say with corals, you know, never touch the coral, but as a scientist, if you're trying to work out what things are, first rule is always to poke it, see. If it is soft, we can probably leave it, but it does look a little bit like an interesting coral.
Yeah, it's brittle, but we've got it.
Okay, oh, yep, no, yes, excellent. Okay, yeah, that is a coral then. That's, if we can try and get it. Yeah, these mesophotic corals, I have cursed myself for working on them in the past when trying to put them back in the, you know, whether it's in a bio box or whatever.
Tom, is this a snap freeze or ethanol?
This guy can just go in ethanol. So yeah, most of the work we do with the DNA, we can just keep the corals in ethanol. It's not particularly important that the tissue is preserved perfectly, but for some of this genomic stuff, yeah, it's really important to snap freeze the tissue as quickly as we can.
Tom, you may know this. I got a question from Jason. Do shallow carnivorous sponges exist? I guess we're talking about the sorts of things that Merrick studies, these ones that kind of spear critters on the side and then they absorb them. Oh, we'll lose. Yeah. Hang on.
Oh, good.
We got some of them, but...
Oh, no. Oh, well, let's see how we go.
Any of these other two that we've got in front of us there, Tom? We did get a little bit of that.
No, that's
one.
right. Yeah, that's okay. Can we just zoom out and I'll have a bit of a look and I'll just try and see if there's any of these more shallow water ones around. Like by more shallow water, I mean staghorns. Yeah, look, to go back to the question, I don't know about the shallow water carnivorous sponges. I mean, I must admit I didn't know anything about them at all until we started hearing about them from Merrick. They're incredibly cool. And obviously in the deep sea, I guess, you know, it's possible you might see them in caves and things like that. But yeah, certainly not something I've heard of. But my knowledge of sponges is not great. So just because I've heard of them or not heard of them is certainly not very strong evidence that they don't exist.
Okay, no worries, you're off the hook. Plenty of sponges here though, really like big vase sponge up on the top left here. Quite beautiful.
Yeah, there certainly is. And Merrick could be right in his element here with all these octocorals and sponges. Some of it's starting to get into these larger encrusting corals. These guys will probably be quite hard to collect. We do see a lot of these again in this sort of habitat. Particularly, we saw a lot around Hydrographer's Passage and things like that. Okay, let's have a look up on top of this ridge. There might be all sorts of interesting things up here. Let's have a look. Wow, it's beautiful. This is in terms of corals. This is really a habitat that's usually pretty rich. You usually see a lot of stuff up here where there's lots of plenty of light available. It's certainly very coralline algae dominated, and some of this, I don't know, this sort of green slimy algae, whether it's Cladomorpha or something like that.
Yeah, just suddenly noted this sort of frond-like, perhaps brown algae, I'm guessing. You know, it's become quite bushy, very prominent all of a sudden on this this 76 metre depth.
Yeah, absolutely. This is, I guess, is this sort of what we would have called the brow at Ribbon 5, Rob? This looks like maybe this sort of feature.
Yeah, I think so, Tom. Yeah. The morphology, we haven't been able to get the multibeam up to really map to this depth zone, so we don't quite know how it sits. But yeah, I think you're talking about that brow-like. Wow, this is a big fan coral in front of us.
It certainly is.
It's like a big scoop, isn't it? Catching all of that current that's sweeping along the edge here.
Yeah, no, absolutely. It's yeah, very impressively large feature. Yeah, these Anella fans are super abundant here. I mean, they're pretty conspicuous. I do notice them when they're around, but you know, a lot of famous dive sites have a lot of these big fans. But yeah, well, there's certainly a really high abundance of them here. None of the corals that I was interested in. There's still some encrusting things, but not the sort of more three-dimensional branching things that I was sort of hoping that we might see. It seems to be very much dominated by octocorals. So in habitats that we get this sort of stuff at this depth, perhaps we can just shoot forward to this next little hard bit and have a bit of a look. But yeah, where we do get this really octocoral dominated system, and particularly with this algae, this really suggests to me that there's probably a lot of upwelling here. So probably getting a lot of variability in terms of the in terms of the temperatures and things like that. Looks like with this algae and things, probably a lot of nutrients as well, relatively speaking, from this cold water. This type of habitat really indicates to me that that's probably the sort of habitat we're looking at. So we often think of deep habitats as being really stable and whatnot, but that's not always the case. So sometimes we can see really large fluctuations in temperature due to upwelling. Sometimes, you know, in the order of 10 or more degrees over a few hours as we get these internal waves that come along and break on the surface. Sorry, on the reef wall. And I wouldn't be surprised at all with this sort of assemblage that if that's the sort of environment we're dealing with. So the organisms that live here on the scale of a few hours might have to deal with temperatures, you know, might be, and varies through the year because the amount of this upwelling varies as well. So we might be, you know, dealing with sometimes temperatures approaching what they are at the surface. You know, we're talking about 20, 25, 28, 29 degrees.
Yeah, we're
and
26
then
degrees
sometimes
now.
within a few hours. So, and then sometimes within a few hours, it might be down to, you know, 12, 14, 15. So it's not an easy place to live in these sorts of habitats.
Yeah, just as far as scale goes, people have a look in the center of the screen. There's 2 green dots are the laser beams, and they're exactly 10 centimeters apart. And so some of those fan corals are well over 1 meter wide. Oh, what's this? Is this a corallimorph?
Someone sampled that coral already. Oh, wow. We're seeing a lot of Leptocerus in here. You can see, even though there's not a huge amount of hard corals, it's pretty rugose, the seafloor here.
A lot of coral here.
Plenty of habitat. There is.
Yeah, you're getting almost a sort of rhodolith. You see these rubble, red coralline algae rubble. I'm just thinking of Coral Sea out in the Queensland Plateau. We came across a Leptocerus zone and a coral rhodolith zone, and then we got a Halimeda zone. It was quite distinct. It was on a very gently sloping shoulder, but we are seeing quite a few corals here.
Can we just, go back down a little? There's a little bit of foliose coral here. Can we just have a bit of a zoom in on some of that? If we go back down just a little bit. Yeah, there we go, getting close. Yeah, not that guy. Just to the right, there's the what looks like mycelium with the big outward facing correlates, and then just to the right, yeah, that one there, to the right of that, sort of just below, bottom right to that, there's actually a fungid sitting on one of those fronds. Yeah, it'd be really interesting to have a close look and maybe collect a little bit of this. If we can go down a little, there's like a pink gorgonian in the middle of it, and there's a frond with a fungid mushroom coral sitting on it. So the really one with all the sort of squiggly looking bits, that's Leptocerus. Probably Leptocerus incrustans like that one. Yeah, okay, can we just maybe, I wonder if that is Parietes reefs. Can we try and grab that piece? It might be Montipora. It's a bit hard to tell from here. Again, we have to look up closely. How do you reckon you'll go at collecting the frond with the fungid on it? That'd be pretty cool.
You want to try and get both together?
Absolutely, why not? Looks like a Montipora from here, but it could also be a Parietes. That'd be an interesting one to grab a hold of.
No worries, we're just getting the ROV sorted here. Looks pretty unstable. We just bumped it, and bits are falling everywhere. So it looks like it's just a big scree slope with some living coral on top of it.
Yeah, it looks almost like someone's already sampled that coral there. Mycelium. Rob, I'm going to have to go in a second. Sorry, it's starting to feel very weird to leave the dive at this point. But I assume, you know, at these delta depths, we're probably getting very close to the end of the dive anyway.
Yeah, that's right. We're at 66 metres, and we've been in the water since 9 o'clock. So it's been a big day for everybody on board. Yeah, so this will probably be the last sample, and once you go, we'll wrap up the dive pretty soon after.
Yeah, no problems. Yeah, I better go now. Sorry, Rob. It's be great to grab that if we can. And thank you very much. This one can just go in ethanol. There's no need to put it in. No need to put it in liquid nitrogen or anything. Just ethanol will be fine. And yeah, it'd be great if we can grab both of them.
Copy that. All
Two
right. Do
for
you just
one.
want to? Do you want to sign yourself off? Just tell people again who you are, there, Tom.
Sure. So Tom Bridge. I'm a senior research fellow at the ARC Centre of Excellence for Coral Reef Studies, and the senior curator of corals at the Queensland Museum.
Thanks very much, Tom,
Right,
and hope to have you join us in future dives as we work our way along Cape York here.
Yeah, likewise. No, very excited to see what we can find. So keep up the great work.
All right. Bye, Tom.
Yeah, no worries. Thanks, Rob.
Anything else while we're in here?
Not that I can see.
Thanks. That's one of the wired ones.
Okay. So we're live. We're good with this at the moment.
Yeah, no, that's all.
Alrighty. Big day. Big sampling day.
Everyone got a lot of sleep last night.
Well, it's 4:30 p.m. here, Marty. I think we've probably had enough time in the mesophotic zone, so I think we'll say goodbye to everybody ashore and bring and recover the SuBastian.
It's been a big day, and we've still got a few hours ahead of us in the lab this afternoon.
That's right. Yeah. So we've got to process all of these coral and the rock samples, the sediment, of course, that we collected right at the very base of this expedition. So we'll just sign off from here. And so this is Dr. Robin Beeman and Marty McNeil saying goodbye from the RV Falkor from the northern depths of the Great Barrier Reef expedition.
#EdgeGBR.
#EdgeGBR, and we are going to aim to do dives every 2 or 3 days, depending on how we go. The goal from here is to continue mapping further north along the offshore part of the Cape York area, this frontier zone of the far northern Great Barrier Reef. So I hope you can join us again on another ROV dive, and thank you very much, and
Thanks,
goodbye.
everyone.